Okay. Greetings and a warm welcome to all the dignitaries present here. I'm Shwastika from Team Clearnet, serving as the allocation assistant to ensure a seamless experience. Clearnet proudly stands as India's most relied upon digital platform, offering a multitude of imaging services exclusively for doctors. It is with great pride that Clearnet serves as a digital partner for this event hosted by Association of Physicians of India, West Bengal. And the focus of today's session is ICTD 2025. Now, I would like to invite Dr. Shambo sir to proceed with the session. Over to you, sir.
Thank you, Clarnet, for being the digital partner of this event. So, myself, Dr. Shambo Samajdar. Currently, I am the Secretary of Physician Research Foundation, West Bengal branch, under the aegis of Association of Physicians of India, West Bengal branch, and Secretary Clinical of Indian Pharmacological Society. So, this International Clinical Trial Day, we are organizing and we are celebrating the theme, Rethinking Clinical Trials, Inclusivity in Practice. So, respected dignitaries, distinguished speakers, panelists, researchers, and dear colleagues. Today, on International Clinical Trials Day 2025, we gather not only to reflect, but to recommit ourselves to the very soul of medical science, ethical, inclusive, and evidence-based research. It was this very date in 1747 that James Lynn, who is a naval surgeon, conducted what is widely regarded as the first controlled clinical trial aboard HMS Salisbury. Tasked with combating the deadly scourge of scurvy, he selected 12 sailors, divided them into 6 groups, and administered different treatments. The group that received oranges and lemons recovered remarkably, thereby birthing not just a cure, but an approach, clinical reasoning guided by experimentation. Nearly 3 centuries later, we continue that voyage, navigating complex diseases, diverse populations, and evolving technologies. But the compass remained the same, scientific integrity guided by human compassion. In the words of Swami Vivekananda, work for work's sake, not for name or fame. Today, more than ever, we must see research as karma yoga, performed not for accolade or authorships, but for the betterment of humanity. Every consent taken, every protocol followed, and every participant respected in an act of seva, that is the service. This year's theme, Rethinking Clinical Trials, Inclusivity in Practice, reminds us the science must be democratic, that the elderly, the underrepresented, the rural, and the unheard must be seen, heard, and studied. True inclusivity in trials is not a luxury, it is a moral imperative. This program, thoughtfully curated with sessions on regenerative research, artificial intelligence-driven trial design, device regulation, infectious disease, and geriatric ethics, brings together India's most accomplished minds from academia, industry, policy, and clinical practice. Our heartfelt thanks to Professor Jyotirmoy Pal, President of Association of Physicians of India, Dr. Nandini Kumar, President of FERCI, Dr. Babul Bezbaruah, President of Indian Pharmacological Society, Professor Santanu Tripathi, President of Association of Clinical Pharmacologists, all our esteemed speakers and panelists joining us today from India and abroad. So before we start today's journey, please allow me to share a personal aspiration, one rooted in conviction and hope. As we rethink our scientific infrastructure, it is my humble dream to see the emergence of a centralized, autonomous, and ethical sound regulatory body for therapeutics. Name could be Bharat, Bharat's Regulatory Authority for Therapeutics and Health Products. Not just an acronym, Bharat represents a vision where India leads not only in the manufacturing of medicine, but in regulating them with wisdom, transparency, and compassion. A regulator that speaks the language of equity, ethics, and scientific pride, true to the spirit of Bharat. Let each of us, as Swamiji said, be great in our place, whether at the bedside, the bench, or the ethics board. Let this event ignite thoughtful dialogue, compassionate research, and lasting reform. With deep gratitude and on behalf of the organizing committee, I welcome you all to the International Clinical Trials Day 2025. Let us begin this journey from the data to dignity, from trials to trust. So it is our real honor now to invite Professor Jyotirmoy Pal, President of Association of Physicians of India, for his welcome address. Welcome, sir.
Thank you, Dr. Shambo Samajdar, Professor Partho Sarkar, Professor Uttam Pal, Professor Santanu Tripathi, Professor Nandini Kumar for taking initiative on International Clinical Trial Day for such a beautiful webinar. Research is the key factor for the development of a civilization. At the beginning of the mankind, of our civilization, it was basically observation based. So diagnosis of diabetes was based on whether the urine is attracted by the ants. So this is the observational power, and on that basis, our science developed. But later on, the modern medicine developed. Basically, it speed up after the French Renaissance. French Renaissance give us a thought that all the knowledge should be based on rationality, based on the evidence. So gradually, we enter in the era of modern medicine. That means there should be some evidence, there should be some scientific basis. So from that particular type of thought, the clinical trial, the concept of clinical trial developed. First clinical trial was conducted in 1940, but that time there is no regulation. So there happened basically now when you want to develop some medicine or some some investigation, human will be the volunteer. But there was no regulation. So unethically, so many people, particularly from the poor countries, from the colonial world, was used as a volunteer. So later on, the Helsinki Declaration make us that the research should be on an ethical basis. And with the in the last 100 years, with the advent of the different clinical trials, so many drugs has been discovered and we got a new life. So in India, we are ahead in different all aspects in technology, in warfare, in space technology, in pharmaceutical, but still we are lacking in the clinical research. So clinical research, that is why API, our organization, is giving due importance, particularly the awareness. The one day, if I think I will be the researcher, you cannot be. So first you have to develop the research awareness and you have to learn the research methodology. Then only you can do the actual research. Whatever the research India we do, many research, many our works is rejected by the international community because of the poor methodology. That is why we always are giving importance at the college level, at the students level. Our organization and we request the colleges, also medical colleges, to conduct repeated courses of the research methodology. If the methodology is okay, then the awareness, then our physician can keep the records, whatever they are practicing in their hospital, in their clinics. And on the based on this data, The time will come when India will make his own guidelines. Till now, we are making our guideline with hypertension, diabetes based on the Western data. But ideally, if ethnic population genetically is different, so we have to make our guideline based on our data. So that is why we need the research awareness. I am very sure such type of workshop, webinar, if happen repeatedly, will increase the research awareness. Along with this, the research methodology, GCP training. So India will enter in an era where the India will be ahead even in clinical research compared to the Western countries. Thank you. With these small words, I wish all success of today's program. Thank you. Over to Dr. Shambo Samajdar.
Thank you very much, Professor Jyotirmoy Pal, sir. Actually, it is your brainchild, so that we have our Physician Research Foundation West Bengal branch. And now, may I now request respected President of Indian Pharmacological Society, Professor B.K. Bezbaruah, sir. Professor Babul Bezbaruah, sir, currently the President at AIMS, Guwahati. So we are really delighted to have Professor Bezbaruah, sir, the President of IPS. So, sir, your welcome address, please. Sir, you have to unmute, sir.
Okay, thank you, Sambhu. It's a nice gesture from the part of IPS, and and especially thanks goes to you for organizing this type of webinar come seminar on clinical trial. As a clinical trial week, you are going to observe, and on behalf of IPS, I congratulate whole team, including you, and hopefully it will be a grand success. Thank you.
Thank you, Professor Bezbaruah, sir. Now, may I request Dr. Nandini Kumar, madam. Madam is President of FERCI, Forum of Ethics Review Committee of India. So, welcome, madam. I request your valuable address during our inauguration ceremony.
Thank you very much, Dr. Samrat, for inviting me to this celebration of the Clinical Trials Day this year. The theme being Innovations and Integrity in Clinical Trials. Every year we have some themes. So this is in keeping with the developments that are happening in the clinical trial area. So on behalf of the Forum for Ethics Review Committees in India, I wish this program great success, and I'm sure that people will benefit from listening to all the talks that have been arranged for this celebration. Thank you very much.
Thank you, madam. Thank you for your kind blessings. And now it is our real honor to have with us Professor Partho Sarkar. Professor Partho Sarkar, currently the chairman of Association of Physicians of India, West Bengal branch. We are really delighted to have with us Professor Sarkar. Sir, can you please tell us about your views on this day?
Thank you, Shambo. I am extremely delighted that our West Bengal API branch, the PRF of West Bengal chapter is arranging in collaboration of the Indian Pharmacological Society and Clinical Pharmacologists Association, such a nice program observing the International Clinical Trials Day in a befitting manner. I welcome all the dignitaries who will be faculties, who will be talking on the subject from 2 to 7. And I think everybody, the listeners, will be very much benefited. As India is lagging behind in all field of research, I think so. So this will go a long way for the future enrichment in the field of clinical trials. And I wish the program is a grand success, and hope if a few of us will get stimulated by all these lectures and will soak in research work, that will be the success story of this PRF of West Bengal branch in collaboration with IPS. Thank you, Shambo. Thank you. Welcome, everybody.
Thank you, Professor Sarkar, for your kind words. Now it is our real honor to have with us our Chairman of Physicians Research Foundation, West Bengal branch, Professor Uttam Pal. Sir, Professor Uttam Pal, we know him, a very vivid researcher. Along with, currently, sir is the Professor of Internal Medicine at Kishanganj Medical College. So, sir, please.
Thank you, Shambo. Thank you for calling me, giving this opportunity to joining today. All the respected faculties, welcome to this International Clinical Trial Day 2025. Today, we are celebrating our work to make medicine better and help patients everywhere. I believe service to man is service to God. I welcome all, and I want this program will be very successful. As the chairman of Physician Research Foundation, we thank you for the hard work and for caring so much about patient. I wish you all a great and meaningful session. Thank you all.
Thank you. Thank you, Professor Pal, for your kind words. And I think there is one small changes in the sequence because we have to match with the US time. We have a prolific speaker from US, Washington University. So Professor Shobbosati Sen. So we had exchanged his session with our own ICMR scientist, Professor Aparna Mukherjee. So the first session from 2:30 p.m. So we will start our academic session from 2:30 p.m. It will be on clinical trials in infectious disease and public health priorities, which will be delivered by Dr. Aparna Mukherjee. And during 4:30 to 4:50 p.m., the lecture will be delivered by Dr. Shobbosati Sen on that stem cells, endothelium, and CVD research. So we will start straight away from 2:30 p.m. now. And in between, I just want to highlight few important points for all the respected delegates who are joining today. Please post your question in the chat box. And we have our dedicated moderator today. The dedicated moderator will post all those questions to us, and then we will ask the respective speaker during his or her presentation. And please be specific on the topic which will be delivered by the speaker. And as we are talking about 2:30 p.m., so we have another 10 minutes time. Take your time. Have a comfort sitting because it will be a long session. I think up to 7 we will have different speakers. We have our panel discussion. We are missing Professor Santanu Kumar Tripathi here in this welcome address because there is some NMC visit is ongoing in his institution. So Professor Santanu Kumar Tripathi, we all know he is a doyen in the field of clinical pharmacology. Currently, sir is the principal at James Medical College, Boj, and sir is the president of Association of Clinical Pharmacologists. We are really proud today that ACP, the Association of Clinical Pharmacologists, also is one of the partner of this event of International Clinical Trial Day celebration. So we will have hopefully Professor Tripathi during our panel discussion session. So in between, we have talks from Professor Aparna Mukherjee first, and then we'll have a lecture by Dr. Shashank Joshi, who will discuss about clinical pearls for mega diabetes trials, what clinicians should know. Then we'll have a talk by Dr. Melvin George. Dr. Melvin George, currently the head at the Center for Clinical Pharmacology at SRM Medical College, Hospital and Research Center, Chennai. Dr. George is also the General Secretary of Association of Clinical Pharmacologists. He will deliver the lecture on From Innovation to Implementation, Clinical Trials of Medical Devices under Indian Regulation. Then we'll have one doctor who is also an artificial intelligence expert, Dr. Santosh Dixit. Dr. Santosh Dixit is currently Chief Domain Expert at Healthcare and Life Sciences Innovation Labs, Persistent Systems, Pune. He will deliver his lecture on smarter trials with artificial intelligence, accelerating discovery, protecting trust. Next, we will have our transferred session, that is from the first to that position, by Dr. Shobboshachi Sen, who is a professor of medicine at GW School of Medicine and Health Sciences, Washington, D.C., USA. He had a prolific research career in the field of stem cell and regenerative science. So he will tell us about stem cells, endothelium, and cardiovascular disease, translating regenerative research into clinical impact. Then after that, we'll have Dr. Prasoon Chatterjee, who is a professor of geriatric medicine at AIIMS, New Delhi. He will tell us about a very important and neglected topic, that is designing inclusive clinical trials for older adults. Innovations, ethics, and evidences. We all know that one of the very important population, that is geriatric population, they are clinical trial orphan. So how we should design inclusive clinical trials for geriatric population, we'll learn from Dr. Prasoon Chatterjee. Then we'll have an important panel discussion. The topic is the future of clinical trials in India, integrating innovation, inclusivity, and ethical rigor. We'll have esteemed panelists with us today, Professor Nandini Kumar, Professor Santanu Kumar Tripathi, Professor Srinivasa Murthy, then Dr. Santosh Taur, Dr. Priyanka Bosh Bhattacharya, Dr. Anirban Rai Chaudhary. So we'll have this panel discussion. I shall be moderating the panel. So after that, we'll have the valedictory address and take-home messages, and we will be privileged to have with us during that time Dr. B. Kalakumar, General Secretary of Indian Pharmacological Society, and Dr. Ashish Shaha, Professor Ashish Shaha, General Secretary of Association of Physicians of India, West Bengal branch. So with that, we are going to start today's event, academic event, by another 10 minutes of time. It will be started dot on 2:30 p.m. And I request you all to be with us. We'll start our scientific processing soon. Welcome, madam. So we had started, we were waiting for you, madam. So again, good afternoon, you all. So we will start today's technical session now. I think I am audible. And to start with, we are really delighted to have with us Dr. Aparna Mukherjee. Dr. Aparna Mukherjee is Scientist F, clinician, scientist fellow, in charge of clinical studies and trials unit at the Division of Developmental Research, ICMR, New Delhi, Department of Health Research of Ministry of Health and Family Welfare. We are really delighted to have Madam with us. I request you all to post your questions on the chat box, and we will take those questions at the end of the Madam's presentation. Madam will tell us about the clinical trials in infectious disease, public health priorities. Welcome, Madam. Now this stage is yours.
Thank you for your kind words and thank you for inviting me. I'm sure you have a very interesting and learned audience who we are being able to reach through this. So I'll share my screen. Can you see my screen?
We can see your screen, Madam, but the presentation I cannot see.
Okay, I'll just stop
Coming.
sharing.
Yeah. No, it was coming. Your presentation.
Okay, so maybe the net is a bit slow. I'll just get it done. Now you can see it?
Yes, it is full screen also, madam.
Right.
Thank you.
Okay, so it's more of, I mean, I have a few slides, but I thought that this would be more like a, discussion regarding clinical trials and infectious diseases. And because the trial environment right now, the ecosystem in India is something which is really blooming and really being encouraged. So we did have a slump in clinical trials, which we witnessed around 2012, 2014, when people kind of, averse to doing trials, as well as participants were averse to getting enrolled in trial because there was a lot of misgivings regarding clinical trials. But as of the current ecosystem, as we can see, is that there is a renewed interest in trials, renewed interest in both conducting, participating, and I would also say funding trials. Because clinical trials, as we know, is the most important cornerstone. It's the best way to gather evidence, whether not only in infectious disease, in any disease that you think of. So what is the scope of these studies that we have in infectious disease? We could have diagnostic studies, trials of a lot of diagnostic algorithms, diagnostic technologies, and we also know medtech is something that is being hugely encouraged in the research ecosystem right now. So infectious disease diagnosis is again one of the main pillars of these medtech innovations. So they need to be validated, they need to be studied. Then we have a whole plethora of preventive, preventive strategies, be it again, bundles, sepsis bundles or interventions which are behavioral, or we talk of vaccines. So in infectious disease, we have a full spectrum of preventive therapy that we can work on. And of course, we have the therapeutics, which could be small molecules, which could be biologics, which could be our, immunoglobulins, etc. So we again have to have a whole spectrum of therapeutics where we are looking into the safety and efficacy of these therapies, if it's a innovative drug, or we are looking into repurposing it for a new indication. And at the same time, we have all the phases that we can go through. Phase 1, where you are primarily looking into the safety, looking into the first in human studies, and up to the second, third, the more definitive trials that do. And the current buzzword in the pharma industry is whether we can actually interpret something from real world evidence. Because as we know, there are number of patients that we treat with many of these, when we are talking about off-label treatment or we are talking of repurposing drugs, we do have a plethora of information with us as clinicians, as treating physicians. So there is a renewed interest as to whether we can use this real-world evidence in a systematic way, in a statistically understandable way to actually come to some conclusion. So these are the various ways that you could do clinical trials in infectious disease. And as you all are aware, COVID-19 has been a watershed moment in research around, be it India or globally, particularly infectious disease. Like if you just see, this is a paper that was published in 2019, that was just before before the pandemic struck, and they did a review of all infectious disease related trials that were registered in clinicaltrials.gov in the last decade from 7 to 17. And they found that ID-related trials only represented 6.9 to 9.9% of the trials that were registered, and that was kind of plateauing over the decade. It's not like it was increasing. But then COVID hit, and we saw an exponential rise in trials related to infectious disease. I don't think so much of research had ever been done that was done in those 2 years. But that did leave us with a lot of learnings and a lot of, lot of warnings as to how we should not do research, and at the same time, a lot of lessons and learnings as to how we can actually better our research. So if I can take you through what are the challenges of clinical trials in ID? Like I just thought that clinical trials is something that you know of, you have already heard of, you might be doing, you might be conducting, reviewing, being part of. But so what is so special about clinical trials in infectious disease? Because infectious disease, as we know, is a major chunk of the patients that we see in India still today. Even if NCDs are rising, they are causing large chunk of the mortality, and morbidity is the main reason of mortality in many of the states, but still infectious disease is there in our daily practice. So what is special about doing studies? But as we saw in that decade-long systematic trial, a systematic review, trials, though it's a high burden condition in LMICs, the proportion of studies, or I shouldn't say studies, the proportion of actually clinical trials that are done on infectious disease are limited. You'll get more trials in the non-communicable disease sections, diabetes, hypertension, etc., than you will find in, ARI or diarrheal diseases. Though you will find a chunk in high-profile diseases like HIV, hepatitis, etc. So what are the challenges? One is that it's a dynamic field. You have emerging, re-emerging infections, right? Though in our country, we cannot call TB, dengue, malaria as either emerging or re-emerging. They've always been there, and now we are fighting to kind of eliminate them. But we do have a lot of other diseases like Ebola, like COVID-19 that we had, other vector-borne diseases. So there is always a cycle where you get newer diseases, less known diseases, which we have to work on. And then definitely we have these outbreak situations, pandemic situations, which in itself has a lot of roadblocks and difficulties in doing trials. And the other way is that if you're talking about a pathogen which causes an outbreak, say Ebola, you have a vaccine for that. Now, where do you test it, right? So you can test it. You can go to the endpoints of immunogenicity, reactogenicity, but you will never be able to go to the clinical endpoint of whether it is preventing the disease or not. Say we have a vaccine come. We can go to the endpoint of having, the neutralizing antibodies, the immunogenicity, but we will not be able to go to the clinical endpoint to say that whether it's actually preventing the disease or not, isn't it? That is a very big challenge in these kinds of diseases, which occur only in outbreaks and are not endemic, will not be found in general population. Then again, as I said, that there is a high burden of infectious disease in LMICs, but then we are very fortunate now that we do have government funding. We have a lot of other avenues of funding in India. But if you talk of the global funding, the funding that comes from the North, the global health funding, as we say, they don't have the burden of infectious disease, right? But the burden is in our countries. So the ideas have to come from us in order to do trials in infectious disease rather than from the global North. So even if you talk of the pharma industry, which is a major driver of such clinical trials, they have less interest in, say, newer antibiotics. I mean, a new antibiotic, there's been ages since we are having newer antibiotics in spite of AMR that we are seeing. In cases of neglected tropical disease, pharma industry has got very, very less interest in this. It's the philanthropic organizations, research organizations, which mainly drive the research here. So there is a lack of funding opportunities, but again, this is changing in time. So what other challenges would you face in clinical trials when you do it on infectious disease? Of course, you are dealing with a vulnerable population, even if it's not an outbreak, if it's an endemic condition. Usually we would find that the extremes of ages are affected badly. Immunocompromised people are affected badly by these. So all these groups in themselves are vulnerable population, and you have to take extra precautions, and you have to be more careful when you are dealing with this group in terms of taking consent, in terms of taking assent, in terms of dealing with their complications, with their adverse events. So in general, you will be dealing with vulnerable populations more than in other streams. And of course, since you're dealing with infectious material, you are dealing with patients who can potentially infect you. You have to follow all the universal precautions as well as pathogen-specific precautions, like we saw during the COVID-19. Research was difficult because of limited access to the patients. You had to have all your protective gear, and only then you were allowed. And even then, the access was very, very limited at least during the initial phase. And then there is always a changing pattern of the pathogens. That is true for all diseases. Even diabetes has kind of, you would say, evolved over generations. But then this kind of a mutating strains that you find in case of HIV, the antigenic shift that you find in influenza, it's very, very characteristic of infectious disease, where actually testing vaccines, testing newer therapies are very challenging because of this mutating habits. Another very interesting point that you should know about infectious disease is the endpoints, because when you design a clinical trial, of course, asking a good research question is the cornerstone of having a good study, right? And one part of that research question is, how do you measure the outcome? What is your outcome measure? What is your endpoint? So usually in infectious diseases, when we are, testing a vaccine or testing a new drug or even a repurposed drug, many a times we depend on immunological, in these surrogate endpoints, of protection or of efficacy. It may be immunological endpoints, whether the antibody titers are rising, or microbiological endpoint, whether cultures are becoming negative. You have early bactericidal action in case of TB drugs. But we have seen that in many cases, even if these endpoints are reached, it may not always translate into actual cure of the disease or a improve clinical endpoint. So what would be more beneficial or of interest to both the physician and the patient would be a clinical endpoint, an endpoint which actually shows an improvement in the patient. It could be a reduction in mortality, it could be reduction in hospitalization, reduction in the symptoms. But those endpoints are usually difficult to achieve. They take longer time. You're doing a TB disease. If you want to actually see clearance of X-ray, clearance of all symptoms, you have and prevention of relapse, you may have to give a full 24 months to see that. So in that, usually what we do is we take these surrogate points. But for your research to be meaningful, it is important that you keep in mind that the clinical endpoints, the hard endpoints, which actually will translate to improved practice and policies, are always preferable. And they, even if your study takes more time, is more difficult to do, it's always advisable to do that, because that will be a more decisive study than rather if you just take a biochemical or immunological marker to say that, well, this has worked or this has not worked. Then again, post-trial access, that this is not unique for infectious disease. Post-trial access is something that we keep discussing even in other conditions, like in cancer oncotherapy trials, where we would like to say that if the drug is effective, people who did not get it, maybe were in the control arm, should receive it after the study has been over, or they should get continued treatment after the trial. Same thing is applicable for infectious disease also, particularly in vaccine trials. We need to have a provision that if the vaccine is found to be effective, the control arm, which did not get the vaccine, should actually be vaccinated after the study is over, and you know that this particular modality is effective, is efficient, and is safe. So that is something that should be inbuilt in our, in our agreements and in the way we design the trial. And we, particularly when we are doing sponsored trials, industry-sponsored trials, we need to negotiate with them to say that, yes, post-trial access has to be provided. Community engagement, again, as I said, is not unique for infectious disease. But since a lot of these studies would be done in the community, you're doing a vaccine trial, you need to recruit your healthy volunteers from the community. So you need to have engagement. And that engagement only does not mean that you kind of give them incentives or anything like that. The engagement should start from before you design the trial, that whether this particular trial, many a time scientific questions are asked because we want to ask them as researchers, as clinicians. But we also need, the mindset is changing, and now we also realize that we need to ask the community, we need to ask the participants whether it will be helpful, whether you want, what kind of questions do you want answered, what kind of endpoints would be really beneficial for you. So that amount of community engagement, it's not that common in our country as of now, or I would say it's not common globally as of now, but it's something that we should endeavor towards, we should work towards. And we do have these community advisory boards now, particularly in cases of sensitive, conditions like HIV, or if you are dealing even with TB patients, chronic diseases, forming a community advisory board where you have patient advocates, you have representations from the community is advisable, because they will help you tell how you can improve your recruitment, your retention, because keeping participants in trials, particularly if you have a long follow-up, is a skill set that you need to develop as a trialist, and good community engagement always helps in that. So, I mean, these were some of the challenges that came to my mind and for infectious disease, and I'm sure all of you who work in the infectious disease domain will have faced many other challenges, and you're welcome to add in the chat if any others that you think of. Then we can always come to the pandemic, which I said has been a watershed moment in all our lives, be it as general population, be it as clinicians, or be it as researchers. So there were very pertinent problems that we faced during the pandemic doing research. There was a lot of research done, a lot of resources that were used. And there were good findings. We had vaccines, we had drugs, but there was a lot of research waste also, in the sense that there were a lot of smaller disjointed studies done, which may not have proved decisive, and many of them did not even complete. Like all the convalescent plasma trials that were started in America and Europe many of them could not finish their enrollment, could not complete their enrollment even before a decisive, treatment strategy had arrived. So what were the challenges that we faced? It was a brand new pathogen. We did not know how it worked or what worked against it. So we were learning as we went. So that, of course, that was the greatest obstacle. And at the same time, we needed rapid response. It was spreading rapidly, killing rapidly. So of course, we needed a rapid response. Usually we take a time to design a trial, do a trial, though we would always say a successful trial has two things in it going in its favor. One is timeliness. It should have a timely completion and the quality of the trial. But here, both of these things were like kind of tested and challenged to the extreme. We had to be very fast, and at the same time, we had to maintain quality in very difficult circumstances. A lot of repurposed drugs were being tested because we didn't know which would work, and that led to actually develop, use of a lot of newer ways of doing trials, like we had adaptive trials, platform trials, which were not so commonly used before COVID-19. And of course, the logistics were a nightmare. There was lockdown, there was difficulty in access to patients because the patients were kept separate from the rest of the hospitals. There was difficulty in transporting samples or medicines or kits. So all these things had to be overcome during that period. And at the same time, there was a hesitancy in participation also, because even as general population, we knew that even the doctors don't know what is working or not. So participating in a trial was something that everybody hesitated in, and particularly during follow-up. Once people were discharged from the hospitals, they did not want to come back to a healthcare provider or to a hospital for any kind of follow-up. So follow-up studies with the COVID-19 positive patients were very, very difficult. So these were some of the challenges that we faced during pandemic. But at the same time, we did learn a lot from this pandemic. As I said, a lot of the way that we do research, that we view research now has changed, has evolved because of the pandemic. Collaboration can work so well, national as well as international, that we saw during the COVID time. We had the public, private, all kinds of enterprises coming together to make the vaccine, to make the other therapeutics. And hopefully many of these collaborations have survived the test of time, and they are continuing even post-COVID. Also, we realize the importance of ready networks that can actually conduct clinical trials. Choosing sites, getting them ready, and then starting a trial takes time. So the countries which actually had these existing networks, which they could pivot, like UK, they could pivot their existing network of NIHR institutes into doing the large recovery trial. In days, actually, they could start. So that was very important. And all funding organizations realized the importance of ready network. Then also we got to learn ourselves and got to use these newer techniques like decentralized or virtual monitoring, right? Till now, we would always say that it's a personal monitoring, in-person visits are always better than anything virtual. But COVID taught us to do everything on Zoom, including like we are doing this webinar now. I'm sure before COVID, this would have been an in-person webinar. So we did all our trials, we monitored them, starting from site feasibility, video calls were done to see the site feasibility, audits and regular monitoring, everything was done virtually. So it was like not by choice, but because we had to do it. So a type of decentralized trial system was developed by most of the researchers. And as I said, newer designs like adaptive trials, platform trials like the recovery, they all came into fruition. These designs were there. It's not that the designs were new. They have been there for ages, but they are mainly used in the early phases of drug discovery or in oncotherapies. They were not so commonly used like now we are talking about them so frequently. In fact, our regulators, our ethics committees, they were also not actually equipped to, review a master protocol and see that there would be multiple treatments, multiple randomizations. The whole thing took some learning on the part of both the reviewers, our regulators, as well as the researchers to get it going. But we can say that post-COVID, we all are more experienced trialists. We have these newer collaborations, newer tools in our hand to be able to do more effective and robust trials. So since we're talking of priority health topics, so infectious disease prioritization is done by many of the funders because we all know that resources are limited. There's only so much resource that can be spent on research. And so we need to know what are the pathogens that we need to focus on, right? So most of the research organizations, funding organizations do their own prioritization exercises. Like WHO, their R&D blueprints, they update this to recognize the pathogens which have epidemic potential. At the same time, where they don't have enough countermeasures so that more research is needed. So this is the present list that they have. COVID-19 is still there. Then we have Crimean Congo hemorrhagic fever, Ebola, Lassa, MERS and SARS. Nipah virus is there. So, we are trying to work on Nipah viruses. Rift Valley fever, Zika, we're also trying to work on Zika virus. And disease X is obviously the probable disease that might cause the next pandemic. So ICMR also has its own list of national health research priorities, which has been developed with a prioritization exercise and which matches with the national health priorities of the government of India. And there are about 11 to now we have increased it to 13 priorities, out of which infectious disease related ones pertain to antimicrobial resistance, One Health, tuberculosis, and all the vector-borne diseases. So obviously the focus are on these infectious diseases. Wellcome Trust, which we know is a important funder globally, also they have their prioritization list to focus on drug-resistant infections, including tuberculosis and fungal diseases which are impacted by environmental changes like the arboviruses, schistosomiasis, cholera, etc., and of course, AMR. So these prioritization lists, as researchers, they help us actually to see where the focus of the global community or the national community is, and to align our research priorities, if possible, to these health priorities. Lastly, I would say that the clinical trial ecosystem in India right now is quite conducive for trial conduct. So it's a very good time to be a trialist in India, actually, because there is encouragement from funding agencies, from government to actually conduct trials, because we have realized that we are a huge population. We have diversity in population. We have the load of the disease, and we have the need to find our own answers. But A very small proportion, about 8 to 9% only of the global clinical trials are actually done in India. And there is no reason why we can't change that. Not only because it's like we need to be a part of the global research system, research community, but then that way we will be able to find answers for the solution, for the questions that we can ask for our own problems, rather than depending upon the global north to ask the questions and give us the answers. So right now we have these enabling, rules and guidelines. You all must be aware of the new Drugs and Clinical Trial Rules 2019, which is a very comprehensive document telling us how to do the phase 1, 2, 3 trials, what are the submissions required. And we now have a very facilitative regulatory agency also, in the sense that they are trying to kind of shorten their approval times. They have a fully electronic submission, which takes away all the need for up and down, coming to Delhi or submitting, etc., etc. They have regular SCCs. They are trying to increase their pool of experts so that the newer therapies like stem and cell and gene therapies or newer biologics can also be evaluated properly. And for example, they have ICMR has deputed scientists from our cadre to the CDSU so that to give them a boost for their SECs also. Then we have a very good document, which is from ICMR, which is the Ethical Guidelines for Biomedical Research and Human Participants, which tells you a lot about how you can conduct studies ethically. Because after all, we have to remember that we are dealing with human participants. So however interesting a question may be, we have to weigh it to the risk-benefit ratio, and the benefit should always be more for the participants and the risk. So this is a very important document that you need to follow if you're a trialist. GCP, of course, the ICH has just undergone a revision. We have the R3, which has taken into newer aspects like risk-based monitoring and more of decentralization, etc. There is an Indian GCP which is published by CDSCO, which is quite kind of dated, but then it has been recently updated. I'm sure the updated version will be published very soon. And as I said, there is a renewed focus on trials in terms of the funding agencies, in terms of the interest in the government. So grants like, as you must be aware, even ICMR grants, now there is a huge focus on finding solutions rather than only describing the problems. So trials get a lot of grants. DBT also has their own networks and provisions of giving grants, particularly to innovative drugs. Then coming to networks, as I said, COVID-19 showed us that we needed networks. We needed ready sites which can work as a plug and play solutions. Whenever we have the need, they can start a multi-center trials. So ICMR has developed a network where there are 4 phase 1 centers, which is a very unique endeavor from the part of the from our part, so that we are having phase 1 centers in the public sector. Then we also have a 75 site strong intent network, which is primarily for the late phase trials. And besides that, as I said, DBT also has their own networks, and disease-specific networks are always available to you. Then we have provisions for CTRI registration, which is the Clinical Trial Registry of India, which is WHO compliant, and it is essential for your publications, for getting grants, that all your trials are prospectively registered. That is, the registration is done before the first patient is enrolled. If you try to register retrospectively after your study has started, that is not possible at this time and date. And I would also request researchers who actually register in CTRI to complete their registration, in the sense that when the study is over, you should upload your reports, you should upload the status of your recruitment, because this is basically a archival, it's basically a data mine for everybody to see what trials are going on in a particular field. And if we have the complete result, that will tell us as to whether the trial was successful, whether it should be repeated, whether we need something else. So in order to have that transparency, it is very important that we kind of upload the results also. So this was in a nutshell that we had regarding the infectious disease ecosystem in our country. Thank you. And if there are any questions, I'll be happy to take them.
Thank you, madam. Thank you for your great deliberation. I think two important take-home message, what I gathered, and I want to tell all the respected delegates who are attending. Many of you are postgraduate students of medicine or pharmacology. Madam had emphasized on the role of decentralized trials. So it is very interesting and important options for us to become clinical trialist. So all of you, please try to learn about clinical trials. It's A to Z. And I think I have one specific question to you, madam. Before that, I want to extend my heartfelt congratulations to you for your leadership in the landmark Placid trial, and it was being published in BMJ. I think India's first multi-center randomized control trial on convalescent plasma during the COVID-19 pandemic. I think three important points what we get from Placid trial set that set an important benchmarks. Number one, it was a pragmatic real-world clinical trial design across public and private hospitals. Number two, rapid implementation under emergency health conditions. And number three, evidence-based reassessment of a widely used therapy during a global crisis. So, madam, my question is, what were the core design elements? I think this could be a very bigger answer, but you can give us one summary. That what were the core design elements of this Placid trial that made it feasible across almost 39 or 40 diverse clinical settings in India? And how can this be replicated in future pragmatic trials?
Right. Thank you for that. Actually, Placid was the first trial globally which was completed on convalescent plasma. As I said, during that time, it was very difficult to enroll patients. So many of the trials that started could not complete the enrollment. They had to kind of abandon the trials in between. So this was the first one which was completed within time. So one, definitely it was because of the emergency, because of the outbreak, all the medical colleges, investigators, they kind of showed interest, they came together. But as I said, this kind of a network is very important. If you have the existing collaboration where you know that these centers are capable of doing a phase 3 trial, then it is much easier to start a trial rather than if you have to start from scratch and identify centers across the country which have the capacity, which have the manpower and the logistics to do a trial. So that is one of the most important takeaway that we had from the trial, COVID trial, and the other, not only Placid, all the other COVID trials, and because of which we really started our phase 1 as well as phase 2, 3 networks. And definitely, as I said, it's a very simple randomized controlled trial, which kind of looked into convalescent plasma versus standard of care. But the essence was to finish it in time so that we have some answers, while it is still important to have the answers. And the essence was to actually coordinate all the 39 centers. So that's where our decentralization or virtual monitoring and that kind of coordination helped a lot.
Yeah, thank you. Thank you, madam. One question I just want to ask, because that has been in our chat box. I think as we were discussing about clinical trials in infectious disease, specifically, what is your take on CHIM model? CHIM, we all know that controlled human infection model studies, CHIM studies. So what are the specific or scientific justifications for using CHIM models in accelerating, especially in vaccine development for different endemic disease endemic infectious disease in India. What is the current situation, madam, here?
Current situation is we do have a guideline, ethics guideline as to how to conduct these, but it's not that we are actually implementing it regularly, right? Because it is a very difficult model to follow in the sense that actually causing the disease, causing the disease in healthy volunteers, and then trying to see there are always two sides, two schools of opinion, which would always argue whether this is at all needed or not. But yes, you have to do it in a setting where you are ready to you have countermeasures for whatever is the fallout of that disease induction, and if your therapy doesn't work, if your vaccine doesn't work, then you should be able to, counteract that measure. So that provision should be there. Otherwise, embarking on this is very difficult. As of now, India, it's a theoretical proposition. We really don't have trials that are using this model.
Thank you, madam. I think we don't have any more questions. Let me just check once again the yeah. So there is no further question as of now. I request all the delegates, if you have any question, you can type that and we can ask madam via email, and we also can get back to you all if the madam will give us
I'll
the
be
answer.
happy to answer.
Yeah. So once again, thank you very much, madam, for giving us your valuable time and for this enlightening lecture. I think many of us had a huge benefit upon your lecture. So thank you, Professor Mukherjee. And now we are moving towards our next session, and that is on a little bit clinics part. So we'll learn from Dr. Shashank Joshi, who needs no introduction, Padma Shri awardee. He's a very senior endocrinologist at the Department of Endocrinology, Lilavati Hospital, Mumbai. So Professor Joshi will tell us about clinical parts for mega diabetes trials, what clinicians should know. Actually, Professor Joshi is currently traveling. He just present at ACE, American Academy of Clinical Endocrinology meeting. So he had sent one recorded lecture for us, and then we will have some question and answer. So may I invite Professor Joshi virtually and let the video on.
So today we are going to learn about mega trials in diabetes, what clinicians should know. I don't have any duality or conflict of interest for this talk, but this topic is very important and relevant because outcomes RCTs teach and rapidly disseminate what can be done, what cannot be done in prevention or treatment for diabetes outcomes. But they must be interpreted in the context of all that has gone before, because they will impact lives of people with diabetes and make them live longer and live with lesser complications. So we all know this from the blood pressure data, that natural history of a 35-year-old white man untreated with blood pressure dies at 76 with 120/80. Will die at 72 with 130/90. Will actually lose 4 years at a little higher blood pressure. Will die at 68 if the blood pressure is 140/98, which means 8 years of life. And will die at 60 if the blood pressure is 150/100, which is 16 years are lost. Which means we lose years if we don't control them well. So, we are always in a war. And in type 2 diabetes, we are winning the battle but losing the war. And that is what Professor Venkat Narayan said in his Kelly Voss lecture. That the evidence that we are winning the battle is we're improving risk factors with people with diabetes, declining complication rates. But the evidence that we are losing the war is that higher incidence of diabetes and increasing burden has been a huge challenge. And therefore, when large outcomes or RCTs, they impact patients, HCPs, and societies, there is a biology to justify any effect. Non-randomized studies are confounded. Small RCTs are not designed to our CV outcomes or our child's findings or bias assessment. Meta-analysis of small RCTs of outcomes magnify problems which are seen in small RCTs. And large RCTs are big enough that all the randomization equally distributes all confounders into two equal groups, of which one gets treatment, and enough outcomes occur in each group to be confident. So therefore, there are levels of evidence from level A, B, and C, class 1, 2A, 2B, and 3. And therefore, there is a evidence-based recommendation system. So clearly, in the today's talk, I'm going to emphasize the role of DCCT in tight glycemic control, type 2 diabetes prevention, care and remission, in prevention of DPP, Finnish diabetes trial, Chinese, Japanese, and Indian diabetes trial, type 2 diabetes in UKPDS and their legacy, the big three trials of ACCORD, ADVANCE, and PADT, the game changer trial, EMPIREC, and then the direct trial for remission. So clearly, DCCT showed us that there is a vascular legacy and tight control matters because microvascular complications are prevented. But the trade-off is hypoglycemia. So DCCT trial was the first trial to show that you can impact there. And when you follow up the DCCT trial, you see a legacy effect on a metabolic memory. And a similar thing which has been seen in type 2 diabetes in mega prevention trials is lifestyle works. So in the prevention mega trials, the landmark trials show us that the DICHING, the DPS, the DPP4, that lifestyle impacts interventions and reduce type 2 diabetes risk across diverse population. They have a global impact on national and type 2 diabetes prevention programs. Their benefit is sustained and the real world implementation remains a challenge. And therefore, we need to diversify the strategies. UKPDS like DCCT was another game changer trial, which was done at the backdrop of the 1960s controversial UDGP program, which apparently showed that tolbutamide had excess mortality, which gave a bad name to sulfonylureas, and excess all-cause mortality with fenfluramine. Unfortunately, that was not true. And they got a small aid from a Clock Workers Foundation, and UKPDS commenced in December 1977. And that trial with simple primary care, led by Professor Turner and later on by Rudy Harman after his death, clearly showed three areas: first phase, second phase, and third phase. And all that it showed us is tight glycemic control, independent of ethnic group matters. And an A1C drop of 1% will reduce diabetes-related endpoint by 12%, microvascular disease by 25%, myocardial infarction by 16%. And this data was consistent and it was persistent. And it very, very clearly showed us that there is a legacy effect. And when we saw the follow-up of DCCT trial, you can see even after 30 years and 45 years of DCCT trial, there is a legacy effect which is sustained and persistently seen. And that data is so clear-cut. But however, that legacy effect is not seen with blood pressure monitoring. So clearly we can see that intensive glycemic control matters and it does reduce both all-cause mortality and myocardial infarction. And after 44 years of follow-up, you can see that the legacy effect is persistent. So when you see UKPDS at 2030, the mean age at 2030 of those patients will be 90 years. And you can see that in 2022, only 14 patients of original patients had an impressive follow-up of 40 years of life. So legacy effect matters. And this glycemic legacy, hit hard, hit early, actually, if you hit hard in the diabetics life, the first decade, it matters. That's what the mega trial showed us. So diabetes glucose control matters, blood pressure control matters. The blood pressure control will have a legacy effect. However, the blood pressure control will not have a legacy. So whether it is type 1 diabetes or type 2 diabetes, DCCT, headache, and UKPDS and follow-up demonstrated a metabolic effect. And this metabolic effect clearly is demonstrated that it will cause, when you hit hard, hit early, functional changes and structural changes and metabolic memory. And this metabolic memory is preventing complications. Here you can see by various plots that if you hit hard, hit early, it will reduce the glycemic legacy and it will impact trial. Similarly, we can see that, when you do this other trials, when they hit hard, hit early, late, like ADVANCE, VADT, ACCORD, mainly VADT and ACCORD, they didn't show that legacy effect. So obviously, the other glucose lowering trials had a neutral effect because they had short periods of follow-ups. The endpoint may need revision. They were resource demanding and costly. study populations with cardiovascular damage, few head-to-head comparisons, and so on and so forth. So obviously now we have drugs like SGLT2 inhibitors. They were not inhibitor there then. So if you introduce a SGLT2 inhibitor, can it blunt or attenuate or impact or extrapolate the legacy effect? And that is possible, that is feasible. And we have data from EMPA-REG now, which was a game changer trial, another game changer trial, which showed that EMPA-10 or 25 clearly impacted hazard ratios across board, whether CV, death, all-cause mortality, and clearly showed that it was a very, very high impact trial. After the pre-statin era, we had the ACE era, then we have the EMPA-REG era. So these three big trials, the statin mega trials, the ACE inhibitor ramipril trials, and the EMPA-REG trials were game changer trials, and the outcomes were sustained. So clearly, EMPA-REG had all the outcomes of a good RCT. It had a good protocol, clear and clear cut. It had a high retention rate, high adherence rate, high event ascertainment, clear analysis presentation, and definitely raised more questions than answers. So it was a good outcome trial. And this impacted all guidelines worldwide that for atherosclerotic vascular disease, kidney disease, heart failure, beyond glucose control, this is the second change which a drug like this can do. And that's something which we recognize and we know very clearly. So clearly we recognize that EMPA-REG trials make a difference. Better glucose control can also have a cardiorenal prevention strategy, and that's the need of the hour. The third mega trial which one needs to know is a direct trial. And Professor Roy Taylor showed us that if you just eat less and have a low calorie diet with the direct mechanism, you will improve the first phase of insulin release. And in the direct trial, very clearly we showed that people between 20 and 65, diabetes not more than 6 years, BMI between 27 and 45, not receiving insulin. Clearly, we could see that in 5 years, there was a very good remission and a follow-up which was seen. And we also showed that if you reduce carbs and proteins, you can modify it. We can use technology to do it. And therefore, what do the outcome trials teach us? They teach us very clearly that if you are able to understand them, they clearly tell us what does not prevent or treat diabetes outcomes. They must be contextualized and interpreted for all that has gone before. And they impact people's lives with diabetes and make them live longer and live lesser with no complications. Thank you so much. But my last point is that mega trials in diabetes space, whether it is prevention, whether it is remission, or whether it is reduction of disease progression, they are all here to stay. And therefore, it's important to remember that lifestyle is supreme, but medications do work. Thank you so much for a patient here.
Thank you, Professor Shashank Joshi, sir, for your wonderful deliberation. I think as we have some time, we'll try to summarize this talk. As we had already told that Professor Shashank Joshi is currently at ACE. So whatever we learned from this lecture, just a summary of that particular lecture. The mega trials in diabetes, they are large-scale, multi-center, randomized control trials involving lot of thousands of participants across diverse populations. So there should be a high degree of heterogeneity. These trials aim to produce high-quality, generalized evidences for different diabetes treatment strategies, particularly in relationship to cardiovascular and renal outcomes. So clinicians must understand not only the outcomes of these trials, but also the nuances of trial design, patient selection, endpoint interpretation to apply the evidence effectively in their real-world practice. So if we talk about a few key scientific insights summarizing Professor Joshi's talks, then number one, the CVOTs, cardiovascular outcome trials. So there was a paradigm shift. CVOTs became mandatory post 2008 saga. We know the rosiglitazone saga, the FDA guidance for all new antihyperglycemic agents to ensure cardiovascular safety. Therefore, we got trials like EMPA-REG outcome, Canvas, Declare TIMI 58, and Leader, and so on. Now we are also giving very recently, we get the results of SURMOUNT and all the other semaglutide, oral semaglutide trials also, which have shown that certain agents, especially the SGLT2 inhibitors, GLP-1 receptor agonist, and now the dual GLP-1 GIP analogs like tirzepatide, offer us CV and renal protection beyond glycemic control. Then number two, beyond glycemia, our focus is on organ protection. So the modern paradigm stresses that glucocentric approach, the only glucocentric approach, is outdated. New agents should be selected based on organ benefit profiles, be it heart failure, be it CKD, etc. Now we are talking about MSLD also. For an example, the empagliflozin, sir had taken the examples of EMPA-REG. So empagliflozin reduces risk of CV death and hospitalization for heart failure, independent of HbA1c. Now, third point, diversity and subgroup analysis matter. So mega trials often included tens of thousands of participants, but real world representation. For an example, in our setting, South Asian population, elderly population, women may still be limited. So we need to dissect the trial data with vigilant eyes. So subgroup analysis help assess efficacy across ethnicities, comorbidities, and baseline risk. So these are crucial for countries like India with unique disease patterns. Fourth point, understanding surrogate versus hard endpoints. Earlier trials emphasized HbA1c and microvascular outcomes. Now we are talking about MACE, major adverse cardiovascular events, renal composite outcomes, and mortality. These are prioritized. So clinicians must discern whether those trial endpoints reflect true clinical benefit for their patients. Fifth point, neutral versus positive trials. Don't ignore the neutral ones. Very important, because not all trials show benefit. For an example, EXAMINE trial, SAVOR TB, they showed neutral CV effect. So these findings are still critical for therapeutic safety and help shape the patient-specific drug selection. So if we want to summarize these or translate this into implication for clinical practice, number one principle, personalized therapy, select agents based on comorbidities profile, be it atherosclerotic cardiovascular disease, chronic kidney disease, heart failure, and now we are talking about MSLD. Number two, therapeutic sequencing. Prioritize SGLT2 inhibitors or GLP-1RA in patients with high cardiovascular or renal risk, independent of HbA1c. We got now the EMPEROR reduced data, EMPA kidney study, DAPA CKD results, DAPA HF results. Number three, safety and tolerability. So be aware of adverse events specific to drug classes. We know SGLT2 inhibitors and their risk of mycotic infections, genitourinary tract infections, how we can prevent those. Gastrointestinal side effects with GLP-1 receptor agonist. We need to know this. Then number four, outcome interpretation. This is very important. We need to evaluate the absolute risk reductions. NNT calculation, that means number needed to treat calculation, not just that those relative risk, because this will give us more realistic understanding of the therapeutics which are using. And fifth and most important, especially in our country, cost and accessibility to treatment. So when we talk about accessibility, it covers both availability and affordability. So consider affordability in countries like India while extrapolating those global clinical trial data. So this is very important to know. So to summarize, primary prevention data in lower risk populations, we need to find out. We need to do some more research on that. Long-term microvascular outcomes, how Indian cohort is representing in the global clinical trials. And now we need to integrate real-world evidences with RCT findings. So I think whatever we learned from Professor Shashank Joshi's lecture, this scientific framing will help us translate evidence from mega trials into bedside decisions that improve both glycemic and non-glycemic outcomes in patients with diabetes. So with that, I think we have our next session now. And for that, we are really delighted to have with us Professor Melvin George. Professor Melvin George, currently General Secretary of Association of Clinical Pharmacologists, and he is the head at the Center for Clinical Pharmacology, SRM Medical College and Hospital and Research Center located at Chennai. So he will tell us about innovation to implementation, clinical trials of medical devices under Indian regulations. As due to some inconvenience, we not get Professor Santanu Kumar Tripathi in the inauguration session. And Association of Clinical Pharmacologists is an integral part of this symposium of International Clinical Trial Day celebration. So may I also ask, before his lecture, Professor Melvin George to tell us about our Association of Clinical Pharmacologists, and then, sir, please proceed for your lecture.
Thank you, Dr. Shambo. I'll just try to share my slides first to check if it's working. Are you able to see it?
Yes, sir. Yes, it is
Okay.
full screen now.
Yeah. So at the outset, I wish to thank Dr. Shambo and team for giving me this opportunity to talk about this topic, clinical trials of medical devices under Indian regulations. So before I start, Dr. Shambo said about ACP. I'm happy to tell that Association of Clinical Pharmacologists is a society that is formed a year ago officially, and the main purpose of the society is to take the clinical pharmacology as a discipline, to the next level, especially in terms of training the next generation of youngsters who are trained in the subject of clinical pharmacology, help them to be part of clinical pharmacology consultations in terms of optimizing treatment outcomes, in terms of improving regulatory practice in the context of clinical trials, and many other salient aspects of clinical pharmacology, and take the profession of clinical pharmacology to the next level. So I thank Dr. Shambo and the organizers for giving me this opportunity to talk about this topic. In these few words, we'll start discussing this issue. So this is the picture of MRI scanner, as you can see. But the important thing about this picture is that what is mentioned right below, and that is the fact that this is not just a simple MRI machine, but this is a indigenously developed MRI scanner, which is something definitely we can consider this a big deal in the Indian scenario, because for a long time we have been dealing with Indian medical devices, by and large, has largely catered around developing simpler, non-sophisticated devices. And we have been heavily dependent on the developed market like Western Europe and US for higher grade, higher risk devices. And that is what is now changing slowly. This is something that happened 2 years ago. And in the coming years, with improved changes in the regulations and all that, we can expect this to become more and more common. So, how big is the medical device market in India? No, it is said that around presently it's around 11 billion US dollars, and as I told you before, this is a market that is heavily import dependent. That is almost 75% of the total market size is said to be import dependent. That means we are largely dependent on the outside markets for the medical devices, and indigenously developed devices are yet there are still there is still a dearth of companies which are bringing out indigenously developed medical devices. Approximately, there are 1,000 manufacturing sites in medical device in India. Around, and also along with medical devices, even in vitro diagnostics and in vitro diagnostic kits are also being manufactured, and that also comes under the similar regulations. And this is one sector that allows 100% foreign direct investment. And the price control for most of the medical devices, as of now, it is not that stringently regulated, except in the case of cardiac stents and knee implants. This is the picture, a little dated picture it is, but nevertheless, it brings out an important point, and that is about how much India is dependent on imports with respect to the sophistication of technology. As you can see, as the graph moves towards the right, you can see the level of imports is more. And as the graph moves on the y-axis vertically, you can see the level of sophistication of the technology is higher. So you can see extreme bottom left. This is where we have unsophisticated medical devices, for example, bandages, gloves, suture materials, all these simpler medical devices. You can find it here. And this is where we are not dependent much on imports. But when you talk about really sophisticated things like orthopedic implants or ophthalmic instruments, dental products, okay, and all these more sophisticated devices, or even imaging for that matter, you can see that there is a lot of dependency on the imports, okay, and that is what we need to change. And that is exactly what is being addressed currently by the government of India to see that the market shift happens. So which are the areas in India where currently medical devices are largely being manufactured? If you see, it's largely Maharashtra, Andhra Pradesh, and Tamil Nadu and Kerala, and also from Haryana. On the other spectrum, we also see that occasionally we get news, just like how we have fake drugs, we also have fake medical equipment manufacturing, and occasionally it gets busted, and that is what the regulations is trying to see, that all these practices are minimized. So how are medical devices regulated in India? We'll have a brief understanding of this before we go into the context of how clinical trials with medical devices are done. So for a long time, Drugs and Cosmetic Act 1940 and Drugs and Cosmetic Rules 1945 was what was the rule for all medical device companies. So the same as how drugs were regulated, the same rules applied to medical devices also. There were no separate guidelines for regulating devices. As a result of that, it is very much an open market, and a lot of ambiguity was there for companies as to how to proceed further. And whenever ambiguity is there, it does not make it favorable for industry to come up and set shop. And that is what changed in 2017 when we started having a unique regulations for medical devices, and that is called MDR 2017. MDR 2017 is the Indian regulations for medical devices. Just like how for drugs we have NDCT 2019, for medical devices we have MDR 2017. Those are the rules exclusively for Indian, for all manufacturers, be it global or Indian manufacturers who want to set up manufacturing or who want to do a clinical trial in India for medical devices. All those things come under MDR 2017 rules. And there was some amendment of these rules in 2020. And then apart from this, we also had a medical devices policy in 2023, which is more like a visionary document, which I will talk about towards the end. So for a long time, the original definition of the Drugs and Cosmetic Act also included such devices intended for internal or external use, the diagnosis, treatment, mitigation, or prevention of disease or disorder in human beings, as specified from time to time by the central government as an official gazette. For, as you can see in this slide, these devices, that is disposable syringes, needles, perfusion sets, they were considered as drugs as per the original Drugs and Cosmetic Act. And in 2005, some more devices were added to be considered as drugs. So meaning these were all the devices that had some kind of regulations under the Drugs and Cosmetic Act. There was some kind of regulation in place using the same Drugs and Cosmetic Act. Okay, and all that changed with the medical device rules that came into existence. And the medical device rules, if you see, talks about how the authorities are constituted, what are the classification of medical devices, what are the processes for manufacture, sale, distribution, import, and labeling of medical devices, how are clinical trials to be done. So normally we use the word clinical trials in the context of drugs, but for medical devices, the terminology they used is clinical investigation. Okay, clinical investigation. So clinical investigation, just a fancy word for saying clinical trials for medical devices. Okay, so it is not any way different. It's just that the terminology changes. So a protocol for a medical device is called a clinical investigation plan. Okay, so apart from this, the medical device rules talks also about import and manufacture of medical device, which doesn't have a predicate device. That means there is no existing standard in the market for that particular medical device. It's a totally novel medical device that is coming out for the first time. There is no precursor, there is no similar product of that kind in the market. So in such cases, how it is to be handled. And then it also talks about who is designated as a medical device officer, device testing officer. And it also talks about something called as a notified body. So what exactly is notified body? So notified body is a private agency which has been conferred a status of having the ability to go and inspect device manufacturing plants and give their approvals. So these are independent private organizations that are given the accreditation for going and inspecting facilities and giving the approvals for those facilities, and that is called notified body. And then we have, it also talks about registration of laboratories for testing and sale of medical devices and export of device and exemptions. And then medical device rules also have certain schedules. I will not go into those details. So how do you classify medical devices? Now this is very important, okay, because the rules change based on what class our product is coming under. So we have class A, B, C, and D. If you look at the US FDA and EMA, it's more like class 1, 2, 3, but more or less the principle is similar. A and B refer to devices which are considered low risk or low to moderate risk, whereas class C and class D are devices which have a high risk, okay? And so there is a risk of safety issues are higher, and so it needs more regulation. In general, class A, class B devices are devices which are largely regulated by the state regulatory authorities, whereas class C and class D, because of the high risk, they are regulated by the central licensing authority, that is the CDSCO. Currently, CDSCO does not have a separate office, a separate branch for the medical devices. There is of course a medical device section, but it's largely called CDSCO only, the device section. And if you can see this this table, I just want you to focus on this particular row, requirement to provide data for devices without a predicate device. Clinical investigation not required for class A and B, whereas for class C and D, clinical investigation is required. So essentially what they're telling is, if your device comes under class C and class D, there is a higher chance for the regulatory authority to tell you to provide clinical trial data or clinical investigation data. Whereas for low risk devices like which come under class A and B, it is essentially the not required. Now, how do I know whether my device comes under class A, B, C, D? So there is a notification that is already provided by CDSCO. Apart from that, if you're coming out with a new device and then you have, according to you, it belongs to one particular class. No, you cannot just like that go ahead and project it as a class C or class B device. We would need to get that also approved by the CDSCO, and they need to tell us which class our novel device comes under, if it is a device which does not have any predicate. So this I told you, central licensing authority, state licensing authority, and we have a national accreditation body which certifies the notified bodies on going and regulating sites which manufacture medical devices. And the standards for medical devices in India that we follow is the Bureau of Indian Standards or any other standards. And every medical device company is expected to have something called as a ISO 13485 quality management system. So in order to get the approval of their product in the market, they need to show a proof that the medical device company is having a quality management system in place. So the standards that is used for that is ISO 13485. It is nothing but a standard, that is quality management system, that is in place for the design component of the medical device as well as the manufacturing component of the medical device. So since 2020, the medical device amendment rules have given a clear definition, and which they have said that all devices need to be regulated totally under MDR rules. For the definition is also again given, those which are essentially devices which are used for diagnosis, prevention, monitoring, treatment, or alleviation of disease disorder, and which is not using, which does not achieve the primary action on the human body by any pharmacological or immunological or metabolic means. So if the product is not achieving its action by pharmacological, immunological, or metabolic means primarily, then that product is considered a medical device. And also under which the specific purposes it's mentioned here: diagnosis, prevention, monitoring, treatment, and supporting sustaining life, disinfection of medical device, and control of consumption. So in medical devices also, we sometimes undertake preclinical research, that is preclinical activities, to determine whether the device is safe and effective enough for use with human subjects. So this could have bench testing, which is basically looking at the components of the medical device in the lab to see whether it is safe, and to see whether the technical specifications are okay as per the standards. Sometimes it is possible to use computer simulations of the medical device to test and see how the device is likely to behave in a particular context. And in a few instances, if it is possible to also use animals, then they should be used. And once the manufacturer believes that the device is ready for clinical trials, they must first get approval for their proposed investigation with all this preclinical data. So the bench testing is largely the workshop testing. So for this also, there are medical device companies which are large, which might have its own facilities for doing this bench testing. Otherwise, these days there are also something called as device parks established in different parts of the country, where a small startup company can go and test their medical device. This bench testing process can be done at that device park, to assess the performance and safety of the device. Then we have the technical testing, which is basically the engineering and quality testing of the materials, the individual components, the electronic and mechanical elements of the device to see for its accuracy as well as reliability. And the prototype is usually rebuilt using this different materials. And then we have computer simulations and animal studies, which are done as and when possible. The form and the medical device rules, it uses the form MD 2022, which is the form that is used to conduct clinical investigation. So if you are having a medical device company and you're planning to do a clinical trial, you need to apply using the form MD 2022, MD 22, and send that to the licensing authority along with the registration fee. And the central licensing authority will evaluate the application. And if they find that the data is fit, that this clinical trial, there is sufficient data to go and conduct the clinical trial, then the permission is granted. And this is the guideline for that form MD22. So I'm not going into the details of that. So in drug trials, we talk about phase 1 testing, phase 2 testing, and phase 3 testing. Whereas in device trials, we talk about pilot study, which is also called a feasibility study, and then pivotal study. There are only 2 major studies that are done. So the pilot study is the initial study that is done in human beings to acquire essential information about the device before moving on to pivotal studies. So pilot studies are done in a smaller group of subjects, largely exploratory in nature. Its objective is to find the preliminary safety and device performance in humans, and also to guide device, whether the device has to be modified, whether future study design should be changed, and also to identify which are the patients who are most likely to respond to the particular medical device. It will help in designing the inclusion, exclusion, eligibility criteria for the subsequent pivotal study. And then we have the pivotal or confirmatory, definitive confirmatory study conducted in a larger number of participants, 100 to 300 maybe, to gather evidence of safety and efficacy. This is equivalent to the phase 3 clinical trial that we have with drugs. And for medical device which doesn't have a predicate device but approved outside India, pivotal studies definitely need to be carried out. And prior to conducting a pivotal clinical investigation in Indian subjects, CDSU may require pilot study data generated in Indian population first before it goes to pivotal study. Okay, so this is something that the CDSU might insist. And this is just to show what are the different components of a clinical investigation plan. So as you can see here, you can forget the top part of this, but the bottom part you can see here. Basically, we have to a protocol of a medical device clinical investigation plan should talk about rationale of why we need this particular medical device. What is the need for this medical device? What is the current scenario in the market? Do we have alternative devices available? What is the safety and efficacy of those devices? How is patient care going to be changed because of the entry of this device? All that need to be first proposed. What is the comparator that we are going to assess when we have our medical device? If is the comparator available? And of course, what is the clinical development stage? And what is the design that you're going to use? So traditionally, RCTs are always considered the gold standard for generating evidence for efficacy and safety. But there is a possibility that sometimes you might not have a comparator to begin with, and then you will have to do a single arm clinical trial, and then it will almost be like a single arm interventional study, and then you will not have a comparator. You will have to see the effect after giving the device when compared to a patient's data without the medical device, and define what are the primary objectives, what are the primary endpoints, what are the secondary endpoints, and particularly look at safety endpoints, and is there any other exploratory endpoints that can be had, which are the ideal patients, for whom is this device? Is this device for screening people in the community? Is this device for identifying or diagnosing patients? Is the device being used for treating patients? All those things need to be clarified. And the inclusion exclusion criteria obviously is very important, because we need to find the right people who are going to be needing this in the clinical context, and what is the sample size that need to be considered for providing the outcome, and how long is the study going to be taking place, and what are the statistical analysis plan that you are going to carry out. So by and large, it's more or less like a standard clinical trial, of course, with the particular issues that we are dealing with in the context of primary and secondary endpoint, and choosing which is the most optimal outcome that can be taken as primary outcome that can show good efficacy and safety results. After a device is launched in the market, the regulator might say that since we are approving this device for marketing approval, but we also want you to continue providing us data. And then it has to be done in thousands of participants, where you give data of patients who are using the device after it reaches the market. This to monitor more of long-term effectiveness. Safety data in a larger population is more likely to identify rare events and how the device is behaving when it is used in the general population. All this comes out in the post-marketing clinical investigation plan, and it's required basically by CDSA for optimizing the intended use of the medical device. So the government of India has come out with a lot of initiatives. So I'll try to skip these slides because we already are crossing the time. But essentially what I want to say is that there are several schemes that have been brought out by the government of India to encourage manufacturers to set up shop in India and develop medical devices. So this is a list of some of those initiatives, and some of the states have also come up with medical device parks, which are essentially used to support the startup ecosystem in India, so that more startup companies can come up, build medical devices, so that indigenously developed medical devices can be approved in the market. And one of the important things about developing indigenous medical devices is that if you see the hospital care cost that a patient has to go through, one of the things that a large amount of the billing goes is the equipment that a patient is using in the hospital. And many of these equipments are currently imported equipment, so it's going to be expensive. So the hospital has to get back that return on that investment, and that is from our own patient bills. So if we have indigenously developed medical devices that are manufactured in India, this is going to all reduce a lot. The billing and the financial burden on the patient is likely to reduce. Finally, I'll just say that there is something called as National Medical Device Policy 2023, which has come out, which is a policy that is expected to take up the medical device sector growth from its current $11 billion to $50 billion by 2030. So it has developed, the government has brought out with different strategies so that ultimately there is more access to medical devices which are used more for prevention of diseases, affordable devices, and also developing a system, an ecosystem where we have trained manpower. Currently, there is no course for developing medical devices in India. So to bring out such kind of courses, to bring out such kind of skilled manpower, and also to improve the quality of medical devices, to ensure security of the medical devices, and also patient-centered quality care, and to improve the research and innovation for all these, this medical device policy has come out. So to help, what are the strategies that the government has brought out? Basically streamlining the regulatory system so that there is a single window system for approvals, attracting investments from foreign players, enabling infrastructure, human resource development, facilitating R&D and innovation, and brand positioning and creative awareness creation. All this are some of the strategies to improve the medical device sector so that ultimately we have more of indigenously developed medical devices. So for shortage of time, I'm skipping all these slides. So the Ministry of Skill Development and Entrepreneurship is keenly involved in developing the skilled manpower and brand positioning I mentioned. And Niti Aayog is directly monitoring this using certain metrics. In fact, there is something called as MedTech Mitra that has been developed since 2 years, where ICMR, CDSCO, Niti Aayog, all of them have come together and developed a system wherein if a company wants to develop their medical device in India, once they approach this portal, MedTech Mitra, the MedTech Mitra through the system, ICMR can actually give them support using the intent clinical trial network, where they have hospitals which can do clinical trials. They connect these hospitals to the medical device companies and also give them their academic support in terms of how best to design the protocol and how best to position the product so that the regulatory approvals are also obtained in a timely manner. So to conclude, I will say the medical device industry is a thriving industry. It's a growing industry, and all these new regulations that have come up in recent years is going to augur well for the market to have patients having our own indigenously developed devices. And this is quite a huge development from where we were 30 years back, 40 years back when it was under the Drug Regulatory Authority. And Medical Device Policy 2023 has immense potential to transform the device industry substantially if implemented as stated. So thank you for your patient listening. I'll try to answer questions if there are any.
Thank you very much, Professor Melvin George, for this elaborative lecture. And I think this is very important for all of us who are using various instruments, various devices in our day-to-day clinical practice and research. I think we have one very important question in the from the delegates. May I ask you, just reading out that question for you. As a practicing physician, I often encounter medical devices that enter clinical use through limited post-market surveillance. How can regulatory pathways in India be strengthened to ensure that innovations in medical devices are both rapidly accessible and rigorously validated through clinical trials that reflect real-world usage in diverse healthcare settings? This has been asked by one physician. I think Dr. Melvin George, please.
Yeah. So thank you for the question, Dr. Shambo. So I do agree that currently the system is such that there are a number of devices where the regulator, if the device company is able to convince the regulator that in terms of safety, the medical device that is in question is considered a very low risk device. Now, then they can actually come out of that mandatory requirement to perform clinical trials, and the regulator might say, okay, provide us post-marketing data. So that is the way they can position their product and convince the regulator. And maybe if they are already having a predicate device, all this can be done. But if it is a device that is more like a class C, class D device, it doesn't even have a proper predicate device, then it is very unlikely for the regulator to just like that ask them to do post-marketing studies and to avoid the preclinical, pre-marketing approval studies. So my guess is that the product that they are talking about is something like very, very less chance of having safety issues, a very less chance of any untoward incidents occurring in terms of its patients' therapeutic efficacy and no safety issues.
I think this is again another question which is very much related to this. What about pathology testing kits? Do all manufacturers to get their kits validated? This is the question. I want to again add something to that because many pharmaceutical industry, they are providing us certain free tests through their own machines, through their own, we can say, point of care devices. And they are giving us the HbA1c result, or for an example, LDL values, for an example, triglyceride values, urine albumin creatinine ratio. But the thing is that with that particular result, if we decide our treatment plan, we may land up into certain complications. So how we should safeguard us? How we can balance between that? So please. Sir.
So yeah, thank you for the question. So there are two issues here. So when you're talking about utilization of clinical calculators, clinical ratios, and things of that kind, I believe our a gold standard should be the guidelines, the treatment guidelines. So most of the established diseases, I mean, where there is an established guideline for many of the common diseases, they would have some commentary on what is the current status with respect to utilization of that particular model, that particular risk score. Okay, in such kind of scenarios, We could just go by what the guidelines says. Now, when you're talking about in vitro diagnostic kits, it is a dicey issue because I understand, as you rightly said, there are some players who, even though they do not have a proper marketing license for their product, it is being sold. In fact, sometimes it is, if you look at even this biomarkers that are used for research, they don't need to get any regulatory license for sharing their product. So it is only when it is coming under clinical testing for clinical care, where decision has to be taken about the patient life or his therapy and all that. It is only then they need to get this clinical testing, clinical validation, and such kits definitely require a validation and approval from the regulatory authority. They cannot avoid that. That is why you will find in non-diagnostic kits, which are meant for research, there will be a sticker mentioned on it, not to be used for patient purpose, for clinical care. So that means that it has not gone through the regulatory pathway as of now. It is as of now still under research. So there is a clear pathway that a manufacturer has to go through to get his product approved in the market for clinical testing.
So it is important to check the CDSU approval status if we are
Exactly.
using devices. So that should be the take-home message, I think. Very important. Thank you, sir. Another question, we can take these two questions together. One postgraduate students, he had asked that pilot study and pivotal studies need to be conducted in disease patients only. This is the first part. And
yeah,
then,
so again, it depends on what is the device that we are using in question. So generally speaking, most of the medical devices are going to be used in patients only. So it doesn't make sense to test that product in a group of healthy volunteers. So by and large, you will see medical device, both pivotal testing as well as pilot testing would be done in the clinical context. If you're talking about the preclinical study, that is before it goes to the regulatory authority, we are talking about bench testing and all that. There, you could even consider doing it in healthy volunteers to generate some data. For example, I want to see that after putting all the electrodes connected to the patient, there are no safety issues that are developed. So I fix that in a few 5-10 healthy volunteers and have the device data that is given for getting the pivotal pilot study approval. But largely, by and large, for pivotal and pilot study, we are talking about diseased patients.
Absolutely, sir. And I think we had conducted one small study on one of our cardiac autonomic neuropathy detection devices. They had purposefully told us to take a disease cohort and also the healthy volunteer cohort to check whether the sensitivity and specificity analysis. So, but in large, as you had mentioned, that we have to conduct it in diseased patients only. So now the next question, I think this could be answered in your manner, that what are the sample size in medical device trial?
So sample size considerations are the same as any other sample size calculation for clinical trials. So essentially, we need to know if you're talking about efficacy study, we need to know what is the minimum clinically accepted significant difference between the test and control that you're expecting, and that is the one that is going to drive the sample size calculation formula. So by and large, these statistical considerations of what is the alpha that you're setting, what is the beta that you're setting, all that, the same rules which you use for any other drug trial, that has to be applied even for a device trial. And of course, you have to see the logistics also. If you see device trials, you do not see the numbers that you see with drug trials. In drug trials today, I understand, I think Dr. Shambo knows very well, that many of the diabetic studies, cardiovascular studies, we talk about numbers like 5,000, 10,000, 20,000. Those are not the numbers that you traditionally see with device trials. It's much more a smaller population. So they are also looking at logistics in the end, and they are much smaller trials than the drug trials.
Thank you. Thank you very much, sir. And I think we are really enlightened with your very much important lecture. And I think we all acknowledge that India is the I hub, that means the innovation hub of world. And most probably for 4,000 or 5,000 years, India is showing the world the path of innovation. Now, when we talk about the transition from innovation to implementation, and there is one bridge, and in that bridge, I think this medical device, it is something innovation. So in that bridge, medical device trials or research is very important, which can tell us how we should use innovation and we can implement that in real world practice. And from that innovation to implementation, now we are moving forward to another I, that is intelligence. And we are now going to talk about artificial intelligence. So we will have a very distinguished faculty today with us. We will discuss about smarter trials with artificial intelligence, in the role of artificial intelligence in clinical trials, how it could accelerate the discovery process, along with protect the trust issues. So we have with us Dr. Santosh Dikshit, who is a chief domain expert, artificial intelligence consultant at Healthcare and Life Sciences Innovation Labs, Persistent Systems, Pune. So we are really grateful to have you with us, Dr. Dikshit. Now this stage is yours.
Thank you, Dr. Shambo. Can you confirm if you can see my screen as well as hear me well?
Yes, it is, but please make it full screen.
Yes. All right. So are we good to go?
Yes, please.
Okay. And the rain gods have blessed us in Pune. So I hope there are no disturbances. But what I'll try to do is, I'll try to save my bandwidth by going off camera. But during the question answers, I'll come back on camera just to gain some bandwidth. So is that okay?
Sure, sir.
Okay. So, okay, so Dr. Shambo and all the organizers, I see a lot of distinguished societies come together to celebrate the ICTD. As a clinical pharmacologist, this is something that we all look forward to year by year. And this time I'm happy to contribute to talk about a very important topic that is capturing everybody's imagination right now. How can we disrupt the current state of art when it comes to clinical trials with the help of AI? I'm a clinical pharmacologist. I'm not a practicing medico, but I come with a PhD in the field. And in the last 4 years, I have been dabbling into data sciences and artificial intelligence, keeping the clinical pharmacology passion intact. And there are some very interesting new areas that are happening. I'm going to tell you in the next 15 to 20 minutes as we go further. So before we actually go to my presentation, I wanted to take a review of where the field stands today. What are the headwinds and tailwinds when it comes to current day clinical trials, right? So if you look at the top, there is tremendous increase in the study pipeline. New targets are getting created, new models, new ways of doing clinical trials. Protocols are becoming complex. There is new technology being adopted. There are new designs. There are diversity and inclusion initiatives. And so this is a very dynamic field right now. And then to meet those new changes, there are certain knowledge gaps that are appearing. For example, there are disrupted workflows. There is a need for a lot of human resource that can look at these kind of areas. And then more importantly, from a study sponsor and site point of view, all stakeholders are feeling the heat right now that you are not able to meet the requirements. There are tremendous challenges. There are budgetary challenges. There are data challenges. There are human resource challenges, etc. Right? So the CRO field is always very interesting. We know that there are more problems than solutions in this field. And this being such an important part of the overall drug discovery development continuum, this is one area that we all need to constantly look at, right? And another big tailwind that is happening is the strategic adoption of AI, despite the challenges that I just mentioned. So there is a mad push right now, and I'm using this word very carefully. There is you can call it almost like a hype cycle right now, where AI seems to be like a panacea for every problem that any enterprise is facing right now. But let us try to be objective here, and let us try to see how AI is really going to be integral to the modern day clinical trial landscape. So in any CRO, we are all part of the CRO ecosystem. When you think of a CRO strategy or an AI strategy, there are about 5 tenets that one needs to constantly be aware of. Right? First of all, is there an overarching strategy? Are you really looking at doing AI for the sake of AI, or is there any strategic investment? Do you have internal capabilities on handling this kind of a strategy? Very important. Much before the strategy is also important that do you have a very good, robust data foundation? Because data, as you can imagine, is the jugular vein of any AI. The only good quality data will give you good quality AI. So do you have the necessary attributes to say that you have good quality data infrastructure? Secondly, do you have the technology layer that can deliver the AI that you are looking for? Now, AI is an often misused term. There are multiple nuances to AI. There is generative AI, there is predictive AI, there is machine learning, there is deep learning. Which AI platform are you going to use, right? It cannot be just one size fits all. So you need to understand that aspect. Most importantly, do you have the teams that can implement and adopt constantly according to the need of the hour, right? So this is where a lot of emphasis on skilled human personnel comes into picture. And in order to keep them constantly sharp and ready with business, you need to undertake continuous learning and innovation, right? So any modern enterprise, whether you are a hospital industry, whether you are a provider, or even a CRO industry, these are typically the thought processes that are all pervading right now in the field of AI. And my own organization, Persistent Systems, is not averse to that kind of a thinking. So what we are doing is, we are taking this conveyor belt approach to the life cycle of a clinical trial. We are all familiar with these 3 phases of a clinical trial. There is a startup phase, there is an implementation phase, and then there is a closeout phase. Right? So if I can break down this entire activity into different silos, but when I mean silos, they are interrelated silos. For each of these boxes, you can see that AI can improvise and improve the efficiency of that entire process, right? And this has been documented very well. There are large volumes of evidence now that tell you that each of these processes can be simplified. You can bring time to advantage, you can bring a cost advantage, accuracy advantage as part of the entire process, right? So the current industry, the current CRO industry is completely possessed by application of AI in all of these technologies. But the real question is, where is the bang for the buck, right? Where is the tremendous impact where you want to invest your resources? So where we think there is a tremendous impact are 4 major areas. One is the trial intelligence. And when I say trial intelligence, it is the design of the trial. The next part is basically at the entire life cycle of trial management, where we look at various phases of the trial and what we can do as part of those activities that I described in the previous slide. And more importantly, you see more and more traction in the regulatory submission part, right? We know how important those 4 aspects are. What me and my group at Persistent we are doing is we are innovating in this space, and we are creating some solutions that can expedite these processes that we just talked about, right? So for example, if it takes X number of hours to do the conventional way of a job in a clinical trial, can you do it maybe X divided by 2 or X divided by 3? So that is the thought process. We are trying to bring in time and cost advantage by creating these solutions. Because I have limited time, I'm only going to take few use cases today, the one that are in the green. And I'm going to try to convince to you that this is probably one way to look at improvising on the clinical trial scenario. So let's take one by one. My first use case is what is called as creating smart clinical trials for patient cohort identification. This is especially true in today's world where we are looking at a lot of biomarkers. And these biomarkers could be precision biomarkers coming from genomics, from proteomics, even your conventional biochemistry or imaging biomarkers. And everybody knows the need for stratification of patients. One size fits all doesn't really cut it anymore. At the same time, you need to really be wise about how to select patients. So how can you improvise your current protocols to bring in AI for improvised patient cohort identification, right? So that is a big challenge. And why this is a challenge? Because we know that wrong selection of patients is one of the single most reasons why clinical trial fails. And it also bleeds the trial in terms of the finances and the outcomes and so on and so forth. So our intention here is to try to simplify this process. And we have created what is called as smart clinical trials. If there is any interest, maybe I can show this demo to you subsequently, but right now I'm just going to show a slide where in this demo, what we do is, let me walk you through the diagram. So the intention here is that when you have a clinical trial protocol document, which is approved by the IRB, you will take the eligibility criteria. The eligibility criteria will be parsed. You will look for specific biomarkers, etc. And then it will undergo some kind of digital transformation. These are called as SQL queries. I will not go into technical jargon. But this SQL query is like a vector that can talk to the patient data, which is in the form of your provider site repository. And by doing some kind of cross-matching between the keywords and the keywords in the electronic medical records, you can rank order the patients that belong to the respective category that you are looking for, right? So if you follow step number 1, 2, 3, and 4, I do entity extraction, I do model generation, then I match with the patient, and then I rank order. And this rank order of patients can be made available to any user in the form of a dashboard, where I can look for out of the 1,846 patients, maybe 680 patients, they match to the particular criteria that I'm looking for. Right? So with the help of AI, all of a sudden, a large pool of my randomized cohorts can be then simplified in order to make it extremely collimated for the specific biomarkers that I'm looking for. And this is now getting implemented in a lot of CROs. Many CROs have these kind of services that can be implemented as part of their utilities for providers and a lot of hospitals together. The idea here is that you still need human in the loop. You are not going to obviate the CRC or the principal investigator, but these are enablers. By creating these tools, you can simplify the process of cohort selection. You will still need the conventional way of interviewing the patient, of going through their physical examination, doing due diligence, etc. But the ability to filter off these patients from your large pool of patients significantly saves time and at the same time cost advantage also, right? So this is one way that the industry is looking at creating smart clinical trials. Another very big area that we are seeing tremendous impact is the ability of AI to help in medical writing. And we all know the importance of regulatory submissions, documentation, due diligence, when it comes to CTD documents, etc. Now, everybody here, I believe, knows about CTD triangle. There are various modules. These are ICH compatible modules. Each module is standardized, it is templatized. There is a lot of harmonization in the way the modules have to be documented so that you can have harmonization across regulatory agencies and etc. But we all know how cumbersome this process is, right? This can really be like a huge mountain load of manual work that is required during that entire process. But can you automate this whole process, right? So that is the challenge here. And what is the challenge? You want to have human in the loop who will have control over the first draft of the document. That is the biggest challenge. You don't want a machine to develop this document by itself, because then it will be exposed to a lot of errors coming from the system. You always want human into the system that can look at it. At the same time, you want traceability. You want primary references to where that data has come from. You want to have authentication. You want to have editing capabilities, etc. Right? So these are the typical requirements of AI-guided medical writing. And what we have done is, using these principles, we have created our own solution. Broadly, in that solution, what happens? There are 7 steps in the solution. On my screen, you will first see that these are the documents that I want to select. For example, I could select a CSR, I would select an analytical procedure document or a clinical trial protocol draft. And then I will have the templatized document coming from the ICH, the CTD repository. And this is where the magic of AI starts coming in. So we can intake now data coming from the raw sources, which are the proprietary property of the client. These are data repositories under protection. And then from here, you can only extract the key entities that are relevant for your regulatory submission. You can avoid signal to noise ratio here. And then that can be auto-populated with the help of AI into these templates. So this is done by a principle called as NLP, natural language processing. And you can actually create a templatized document. More importantly, the human comes into the system where the human expert can edit, update, delete, and look at the next draft of document. And then we have also created something called as evaluation criteria to determine what is the quality of this draft, right? And I'll come back to that in a minute. And then if your quality is satisfactory, then the final human user can approve the document, lock it with a signature, and then send further into the regulatory submission pathway, right? So this is how most of the industries are now doing regulatory submission documentation. What is very critical here is how do you determine the quality of the generated document? And there are some routine checks. These are standard industry practices that have come in. So for example, are you adhering to the standards of writing? For example, if you are writing for EMEA, are you following those patterns? Then your general English syntax or your language syntax. Accuracy against the literature. So can you cite the primary references? Very important. And then can you do vocabulary terminology matching? You can do ICD code matching. You can do a lot of these kind of activities. And each one of these can be rated with the help of aggregate scores. And let's say you can benchmark that any score which is 80% and above, therefore is an acceptable document, right? So this is the way you look at quality control into the overall system. For lack of time, I'm going to move into the third slide. And this is a very interesting story. In the new era of adaptive clinical trials, we are looking at new age designs. And there is this big push for replacing your conventional control arm with the synthetic control arm. And the advantages are many. You can have completely look at rare diseases. You can have a lot of data coming from just statistical comparison. You can completely get rid of placebo controls, etc. So many advantages of synthetic control arms. But US FDA or any regulatory agency is asking that the control arm has to be created according to certain standards. And these standards are called as fit for purpose standards, FFP standards. And therefore, the requirement is if you are going to create a synthetic control arm, how do you determine that the quality of that control arm is according to the standards set by the FDA? And this is where there is tremendous impact of AI. You can actually generate synthetic data with the help of AI with a technique called GNN. These are generative neural networks, which can actually create a centralized data according to your Poisson distribution of the original data from where it comes from. I will not go into detail, but this is a very advanced mathematical algorithm. But this works very well. You can create a synthetic control arm. So what we have done is we have taken one of the most complex scenarios of creating synthetic data for medical images. So let's say you are doing a cancer imaging trial with radiology. You can see tremendous impact of radiology and AI. So we have taken this use case. And for some reason, you are not able to meet your recruitment standard. So instead of 100 patients, you were able to only recruit 75 patients. But you still have that shortfall of 25 patients. And for them, we can generate a synthetic control arm with the help of this synthetic data created by the GNN activity. And by doing that, if this data matches to your original parent pattern, according to the statistical distribution, FDA will accept this synthetic control arm, and then you can do comparative studies between your control arm versus your treatment arm together, right? So this is the state of art when it comes to synthetic data generation. Doing synthetic data for numerical or textual is, in fact, an easy challenge. Medical imaging is where there is tremendous need for doing this kind of innovation right now. So this is all pervading right now. You can see tremendous impact in the entire value chain of clinical trials right now. What is the mantra in today's world, right? So the mantra is as follows. Put a lot of premium on your quality of data. So try to get as close as possible to fit for purpose data. So everybody is currently investing in upgrading their data infrastructure, data quality. Secondly, be very smart about using AI because AI can also be a money bleeder. So be very judicious, be very cautious about which AI you want to use where. Most importantly, bring in responsible AI because this entire activity is only as good as the trustworthiness, the explainability, and the intricacies of the AI that you will bring together. And this together will give you actionable insights. I have just cited a few examples where you see tremendous impact of biomedical applications, what we call as actionable insights that are getting tremendously overall because of AI impact. So the entire value chain of a clinical trial, which I described earlier, and then so many functional use cases, you can see now coming under the ambit of AI and clinical trials. And this is only probably this is going to snowball on us, right? So we are not going to run away from this particular activity. But a word of caution, because Dr. Shambo told me to talk about trustworthiness. There are inherent loopholes into the system. And because of these systems, there are risks associated with this whole activity. So what are the risks? One is the data that you are going to use has to be absolutely sacrosanct data. There cannot be patient identifiers. We know the ethics of PHI sensitive data. Therefore, you need to bring in a lot of data privacy security. Many of these models have inherent bias, and that bias can continue as part of your AI. So you want to minimize the bias. Wherever possible, you want to have consent, you want to bring transparency. You don't want to bring over reliance on AI. You still want human to be in control. And even 10 years from today, this principle will remain. And then you want a lot of documentation, you want transparency, explainability, etc. And until you have external oversight, you want internal oversight also, right, to mitigate all these things. So there is a tremendous investment right now happening in responsible AI to identify the risks of these kind of activities and then how to mitigate and control these risks together. So for each of these activities, there are solutions available. You can bring in a lot of good data practices. You can bring fair principles. You can bring enhance due diligence into that entire process. And US FDA, GDPR, European Union, Government of India, almost every regulatory authority is coming up with their own local regulations. So we need to be compliant about all this. And above all, external regulations will not suffice. The humans that are involved in this enterprise have to be more and more diligent. So these are called the tenets of responsible AI. I am going to end with this slide. So this is a top-down, bottom-up approach. The last person in the value chain has to be responsible. The principal investigator has to be responsible. So that means you have to have organizational principles. You have to have constant training. You have to have robust data systems. You have to have frameworks. You have to have reporting standards. You have to have some kind of corrective mechanisms when there is an oversight or a breach. So these are principles that now enterprises are adopting, whether you are a provider or a CRO or a pharma company. Because unless you adhere to any of these principles or all of these principles, you are going to stonewall yourself when it comes to regulatory submissions and realizing the true potential of AI in clinical trials. I'm going to end here. I'm very happy to take some questions as part of this equation, and I'm very thankful to the organizers for inviting me here.
Thank you very much, Dr. Dikshit, for this wonderful deliberation, I must say. And I think the last slide, which is very important for all of us, and one question that had been asked by one of our delegates, that is, what are the models which can be generated by AI? So is it possible to, again, if you just focus on that particular slide, and also if you want to answer it?
Yes, absolutely. So I think this is a generic question. What kind of models can be generated? So let us understand the classification of AI. The conventional model of AI is where you take a learning data set and then you blind that data set, you develop a model, and then you expose a blinded data set to that model where it will give you some kind of prediction. So this is called as predictive AI. But the modern way of doing AI is something called as generative AI. So for example, somebody has already created these models for you. You must have heard of large language models and then large multimodal models. So there is no one rule of thumb here. It depends on the kind of question that you are solving for. In today's world, there is a more impetus on using generative AI. That means pre-baked models. A lot of open source data is available. If you have heard of Google, Microsoft, each one of them are creating healthcare specific models because there is large amount of data available from hospitals. The only downfall of those models is that there is an inherent bias and they hallucinate. So if you can manage the hallucination by bringing some kind of checks and balances, I think you can work with the AI. But if you are looking for 100% accuracy in a predictive model, for example, you are developing a medical device. My previous speaker talked about diagnostic devices. In that, you cannot use generative AI. There, you want to use absolute diagnosis. And therefore, you would rather use machine learning or your predictive AI in that kind of a situation. So it's a double-edged sword. AI model selection depends on the objective of the enterprise work that you are going to do.
Yeah, thank you, sir. One question just came into my mind, that sometimes we encounter patients with overlapping comorbidities and sometimes with incomplete records. So if we can use certain artificial intelligence enabled clinical trial platforms, because we are now talking about pragmatic clinical trials, real world evidence studies, so which could identify certain real world eligible patients more accurately, while also that could ensure the transparency So how artificial intelligence models make those decisions, if that is available, or what is your take on this?
So let's take let's say you are running a hypothetical clinical trial with 500 patients, and you are only able to determine a cohort of 300 patients despite the time limit. And within those 300 patients also, there are a lot of missing gaps in the data, right? So, and this is reality of any clinical trial. So in this situation, you can do something called as synthetic data augmentation. I just referred to it. And if that synthetic data augmentation is done according to the regulatory standards, I think that is well acceptable. You can clean up your data and then try to make it compatible. So there is a term called AI readiness of data. So that AI readiness can be brought in with the help of synthetic data and AI models. That's one. Secondly, almost every clinical trial platform, electronic healthcare record now is AI enabled, whether it is Epic or whether it is a Red Hat or anything. So in that process, what happens is these AI can also determine where are the gaps, and it can propose the gaps that need to be overcome for doing this kind of analysis coming together, right? So the entire industry of electronic healthcare record is also undergoing a lot of disruption because of the adoption of AI. But ultimately, I think the tremendous impact of AI is data analytics downstream, not so much in the operations. So for example, let's take the oncology clinical trials, right? So if in a conventional way, your Kaplan-Meier curve only has let's say a 40% separation between the two curves, the survival curves of the two cohorts. But if AI can tease it further, and it can show you that the true effect is not 40%, but maybe 70%. Just imagine the impact that will have on the marketability of that trial or the overall business impact of these kind of activities. So one of the biggest impact that is happening in data analysis is called multimodal data analysis. What does it mean? You can bring in images, you can bring genomics, you can bring every kind of modality of the data, and you can harmonize that data. And that harmonization is closer to the biological truth or the clinical truth. And by multimodal data analysis, you can actually get into the nuances of primary and secondary objectives. And I think that is where you are seeing tremendous impact on the clinical trial outcomes because of AI.
Yeah. So thank you. Thank you very much. I think we'll search for more questions if it is there. If it is not, so we are really thankful to Dr. Santosh Dikshit for this thoughtful and very comprehensive lecture on how we can make our clinical trials smarter by using artificial intelligence. Thank you, Dr. Dikshit, for your
Thank you.
wonderful deliberation. And if you want to make any one take-home message for all of our delegates, you can make.
Okay, my message is, in the search of artificial intelligence, let not lose natural intelligence.
Yeah, so human intelligence is very important.
Yes.
I think that is the more important statement. So thank you once again, Dr. Dikshit,
Thank you.
for your beautiful lecture. So now we are really delighted to have with us Dr. Sabyasachi Sen. Though here it is afternoon or evening, but we want to say Dr. Sen, Professor Sen, a good morning, because it is 6 a.m. in Washington. So welcome, Professor Sabyasachi Sen, in our platform, where we are celebrating International Clinical Trial Day. So Dr. Sabyasachi Sen is the professor of medicine at George Washington School of Medicine and Health Science at Washington, D.C., one of the premier medical institute in US. So welcome, sir. We will learn from Professor Sen about stem cells, endothelium, and cardiovascular disease, and how we can translate regenerative research into clinical impact. So now this stage is yours. Over to Professor Sen.
Thank you, Dr. Samajdar. It's actually 7, thank God, and not 6 o'clock. but because it's 9.5 hours behind. But thank you so much for giving me the opportunity to talk about endothelium. Let me try to share my slides. I wonder, can you see it?
Yes, sir. Please make it full screen.
Is it full screen now?
I think it is not full screen because in some different modes it is showing. You can go to the display setting up. There is one display setting, I think.
Okay.
You can click on that. I think there you will get the option. Yeah, the swap. Yes, you can try that. Yes,
How-
now it's better. Yes, sir.
It's better? Okay.
Yes, yes,
Sorry, it seems to be listening to something here, but I don't know what it's listening to. But anyway, is this okay?
Yes, yes. Okay, sir.
Okay, good. Well, good morning, everyone, and thank you for giving me the opportunity for giving this talk on endothelium and adult stem cells as a biomarker, not only for regeneration, but also as a biomarker in clinical trials. My outline would be introducing endothelium and diabetes, or diabetes and endothelium, the role of endothelial progenitor cells, a little bit on stem and progenitor cells in general, and then its role in cardiometabolic diseases and its role as a biomarker, other modalities of endothelial function or indirect measurements that we usually do. So I'm going to touch upon on that at the end, near the end of the talk, and summary and conclusion. So as you all know, diabetes is a major disease across the world. Both India, United States, and China still are in the top 3 as nations in the world with diabetes. Both type 1 and type 2 and gestational diabetes are the main types of diabetes. This is a little bit dated slide, but it talks about 422 million adults having diabetes, with 1.5 million diabetes-related deaths. So, but the crucial point in diabetes is the vascular diseases. There's many vascular complications from diabetes. So the reason we treat diabetes is not because of the sugar, but because of the cardiovascular disease or cardiovascular burden that diabetes presents. And the key to that is essentially the core lining of the blood vessel, which is the endothelium. So endothelium has good parts and the bad parts that impacts it. It's probably one of the largest organs in our body that lines almost every blood vessel that we have, because it needs to dilate and let the blood keep flowing and perfuse. So that includes nitric oxide, shear and stress, antioxidants. And I really want to stress antioxidant effect at a cellular level, regular exercise, and healthy diet. Healthy diet, when I say healthy diet, I would stress more so about non-artificial sweetener and non-processed food diet. On the other side, we have obviously anything that is anti of the green side, which is oxidative stress, hypertension. Dyslipidemia is one of the main impacts on endothelium, smoking, and hyperglycemia. So the diabetes-related endothelial dysfunction, yes, there is effect of glucose, but then there is effects of lipids as well. It causes nitric oxide to go down, prostaglandins to reduce, and on the other side, it increases the inflammatory molecules as shown here with angiotensin 2 and COX-related factors. But you have to realize that the main function of vasodilatation and vasoconstriction is happening in the smooth muscle and not in the EC. and there is a balance between vasodilatation and vasoconstriction, which ultimately leads to the diameter of the blood vessel. So this is a little more in-depth into that. As I mentioned before, endothelium is the innermost lining. Then we have the SMC or smooth muscle layer. Then we have the fibroblast. There is, as I mentioned before, the nitric oxide, which is the critical element or function of the endothelial cells, comes from a critical amino acid called L-arginine, which is by action of endothelial nitric oxide synthase enzyme produces nitric oxide, which then transfers in through the second channel through the GTP and GC, and via cyclic GMP causes vasodilatation in the smooth muscles. It has to diffuse down. Nitric oxide has to diffuse down from the endothelial cell layer to the smooth muscle layer to ultimately cause the vasodilatation. And of course, as I mentioned before, the reactive oxygen species is actually important, where if you have too much reactive oxygen species or ROS, that would impair the nitric oxide, leading to peroxynitrate, which does not lead to vasodilatation. So essentially, there is a physiological aspect to the nitric oxide, and then there is pathological conditions such as diabetes, rheumatoid arthritis, any inflammatory condition, which causes so-called the uncoupling of the nitric oxide. The hydrogen peroxide comes in. There is interplay of various genes such as NOXs and causes the endothelial dysfunction. Now, this actually is fairly pervasive, and that's why the endothelium needs to be regenerated at a very fast pace on a daily basis, and that's where the endothelial progenitor cells comes in. These are essentially tiny cells that looks like similar to the upper top part of the slide, which almost looks like a mononuclear cells from blood, but these are identified essentially for progenitor as CD34 positive cells, KDR meaning VEGF receptor 2 positive. So if you ask me, how do you identify these cells in blood or mononuclear population, these are essentially the markers. So you can flow cytometry sort these cells and subsequently study them based on as the need may be, and also to get as close as possible to the endothelium without actually tearing it off of some small vessel in your body. So these are essentially baby endothelial cells that are released from the marrow. The other cells that I'm not going to talk to you about, but are very critical in our body, and that does modify in diabetes, is the mesenchymal stromal cells. These look like the spindle-shaped cells. These are identified from CD73, 105, and 90 as far as the NIH guidelines. And the source could be any mesenchymal tissue, but these could be used as a biomarker as well. And as I mentioned, I'm not going to talk about these cells today, but these do get modified in diabetes as well. So going back to endothelial progenitor cells, as I said, these are the cell surface markers. These are mostly in the marrow, excuse me, and these move from the marrow into the peripheral blood in response to chemotactic factors that are released from a dying or diseased tissue. Only at any particular time, there's only 1% in the periphery, but in times of need, these actually can increase way up to 5 to 10%. So these cells are anti-apoptotic, pro-proliferative due to their secretory factors, but their prime function is migration. Without the migration, the cells will not be rescued, or damaged endothelium will not be rescued. And this so-called homing property is one of the critical functions. And this is sometimes mostly mediated by a very well-known chemotactic factor called SDF1 alpha, or stromal derived factor 1 alpha. And this is critically impaired in diabetes. So though one might actually have enough CD34 positive cells in the periphery, they're not reaching the site of injury or tissue damage. So as I said, these are the cells at day 0 if you take it out of a patient's blood. And these actually do make colonies by 14 days. And as I mentioned before, these are essentially one needs to move from the marrow to the periphery, identified by these markers, and gradually from early circulating EPC or CECs, so-called become circulating EPCs. But as you can see, some of these marks continue to change, and ultimately the goal will be from 34 to 31, which is the progenitor marker is 34 and mature marker is 31. So they would gradually mature and migrate and then hopefully settle down. So this is essentially how it settles down to form the new endothelial monolayer. And as I said, the EPC gradually needs to mature over a period of time. The period of time is usually between 2 to 4 weeks, depending on the need and the number of progenitors one might have. Excuse me. So the EPCs in regeneration has been used over a period of time in various settings, particularly for cardiac regeneration, for heart failure, ischemia, and so on. And the number of cell therapy had increased dramatically across various universities in United States, Europe, and also Asia. We used some of these cells in our mouse model going back almost 15 years now, where we transplanted endothelial progenitor cells in an acute ischemic mouse model, where we ligated LAD, and these over a period of time did help to regenerate the left ventricle. As you can see on the left-hand side, we have the non-cell therapy heart, and on the right side, we have a cell therapy heart, and as you with EPCs, and as you can see, there is well preservation of left ventricular muscle. This is a cross section across the left ventricle in the heart. And we showed that there's actually improvement of fractional shortening, which was as low as 19% to went up to almost 25%. Now you can say, well, this is only 6% improvement, but that 6% actually would change your NYHA classification from class 2 to class 3. And which means that a person can go up the stairs or even 3 flights or not. So there has been multiple human trials using EPCs over a period of time for ischemic regeneration. And as I said, in diabetes, these are the certain factors that impairs the endothelial function, and these have been used for various diseases such as diabetic retinopathy, peripheral vascular disease, and coronary artery disease. These are also the complications of diabetes. And this also summarizes what I've also already mentioned to you about the migration number and functionality. So if one has to use these cells as a biomarker, one needs to look at the number, functionality, meaning the migration, which is actually, as I said, down-regulated, tube formation or colony formation, unit formation, and addition or inflammatory molecules by gene expression. So we, in our studies, specifically focus on diabetic kidney disease. And the reason being this particular complication is actually increasing dramatically, more so in non-Caucasian populations across the world. There's 1 in 3 patients with diabetes complications, and almost no patient that I see has a EGFR that is at 90 or above. So everybody is essentially at stage 2, if not stage 3 CKD. And this is primarily because of the effect of hyperglycemia hyperdyslipidemia, and which ultimately leads to podocyte injury. I'm not going to talk a huge lot about the podocyte injury today, but remember this is a podocytes are very key to maintenance of the called the glomerular function in diabetes. So anything that injures it, particularly the ROS, would lead to podocyte injury. So diabetes is a major cause of endothelial dysfunction and ischemia, and it's primarily leads to poor perfusion. Here is a picture of normal glomerulus on your left-hand side, and on your right-hand side, you have diabetic glomerular disease where there is reduced podocyte number and glomerular basement membrane thickening. So we looked at various drugs that are known to have some beneficial effect in type 2 diabetes as far as CKD. These would be primarily the SGLT2s. We have various SGLT2s, and these are fairly, I would say, in our practice, we try to put it on almost all patients because not only are they anti-hyperglycemic agents, but they also improve cardiovascular structure, cardiovascular function, renal function, perfusion in general. So we did a study using canagliflozin. These are, as you can remember, this drug was fairly first to come out, but this is a lot of what we saw was a class action. So we used a low dose. As you can see, we used only 100 as opposed to 300. We subsequently now are examining empagliflozin at a low dose, but let's go back to this study, which was 16-week study, very short study. So the beauty of using the stem cells is that you can use and keep your clinical trials at a short period, as opposed to several months or even years that you need to get a MACE outcome. So from our outcome measures, we took serum, we took the stem cells where we looked at the migration, colony formation, and so on. And from the urine, we actually looked at the urine exosomes to discern podocyte inflammation. And we looked at specific podocyte-specific proteins such as podocalcin, nephrin, and Wilms tumor. I'm not going to talk about that today. Concentrate mostly on the blood-derived factors. So interleukin-6, as you can see, went down dramatically with canagliflozin, which is the inflammatory molecule. Migration assay improved quite dramatically within 16 weeks. Remember, this is only 16-week study. And CXCR4, which is actually very critical, as I mentioned, CXCR4 is the prime receptor that helps the cells to respond to SDF1 alpha. So the conclusion from this study is that it showed improvement even at a low dose with the blood pressure, inflammatory molecules, and also certain genes, particularly the antioxidant genes. I know I didn't show you the gene expression studies here, but we did actually very recently another SGLT2, but this time we actually had a combination with DPP4 inhibitors. This was recently published in Stem Cells. This was a fairly short, small cohort study with only 4 or 5 patients in each group. But even there, you actually saw a reduction. So the blue bar is the control, red bar is the dapagliflozin, and green bar is the combination. And as you can see, in almost every group, there is improvement in the combination. And however, if you look at the red and the green bar, there's not a huge lot of difference. So dapagliflozin alone, without the SGLT, without the DPP4, is actually doing a lot of the good things that we would expect, such as CRP reduction and so on. Clearly, the lipids are better with the combination. If you go to the previous slide, clearly the glycemic control is better with the combination. If you look at the cell values, again, the migration fraction, which we got the statistically significant difference even at 16 weeks, using a low dose dapagliflozin with the low dose of saxagliptin, we do see a improvement both in DAPA and combination almost at a similar level. The colony formation unit didn't show a significant statistical difference, but the combination was slightly better. I wouldn't put a lot of stress on that, except the migration was clearly a better assay. CD34 numbers, again, were equivocal across the groups. Again, the conclusion from a low dose dapagliflozin and low dose saxagliptin, and as you can see, here is a pattern. The pattern is we have consistently done a low dose SGLT2 studies just to because these can be used not just in diabetes patients, but in non-diabetic patients with CKD and heart failure. So that's why we have kept doing this low dose study. In spite of pharmaceuticals coming back and telling me to do it in high dose, but we thought that you could actually get a lot of the positive effects even at a low dose of SGLT2. And clearly the cellular measures, particularly migration, appears to be consistent and favorable with or without the combination with the DPP4. Moving on to the non-invasive studies, as I mentioned that I'm going to give a brief overview of these. We also look at certain tests such as flow-mediated dilatation. This is fairly well-established study. You use an ultrasound probe with a cuff where you inflate the pressure, and as you can see, you would expect about 11% improvement or increase in the diameter of the vessel. If you give nitroglycerin, which we don't, which a lot of laboratories don't, you can expect up to 18%. In our studies, the FMD was fairly reduced. It did improve a slight bit. Going to the next study, one can do a brachial artery ultrasound as well. And the last study that I would also talk about is a PAT, or peripheral artery tonometry. It's fairly easy to do. I think this should be incorporated in almost every clinical trial that we have. Non-expensive, gives a lot of information about reactive hyperemia, which is important and shows shear stress effect of enos. And another study that we have consistently done in all our clinical trials is pulse wave analysis and pulse wave velocity. This is a good study. This is not dependent on ultrasound and the person doing it. One would ideally, this is essentially looking at central pressure as opposed to peripheral pressure. So we take a measurement at the femoral and the carotid, and then compare the pulse wave velocity and pulse wave analysis. This is one of our studies, very early on studies where we just use saxagliptin. And as you can see, if you just take out the visit 2 between visit 1 and visit 3, there is obviously a progressive increase in the augmentation index, but with saxagliptin, it actually went up and then came down quite drastically, whereas the placebo, it actually stayed fairly high. So we concluded that this saxagliptin patients at least remained blood pressure was stable and not high and low with the so-called placebo, and as you know, that diabetes is a progressive disease. You don't want peaks and troughs. You want more a stable, low blood pressure. So augmentation index actually is a good measurement for it. So the conclusions from this talk one can take away is that our studies with using a cell in diabetic vascular disease monitoring using medications, low-dose medications, SGLT2, GLP-1, and so on, the cellular studies are missing. And I think the main take-home point I would say is we need more cell-based studies beyond serum-based studies that we have. These could minimize the time of our trials and gives a lot of information about individualized response to intervention. And thank you so much for listening to me and for inviting me to give this talk. Thank you.
Thank you, Professor Shen, for this enlightening talk. And I think this is a very important for all of us. We are very much naive to stem cell research because lots of other issues, lots of especially human stem cell research, regulatory issues. I think Professor Nandini Kumar is also there. I think he knows she was long time attached with ICMR, deputy director, and I think there are lot of hurdles with stem cell research in India. I think one question in my mind, if we talk about translating stem cell based research in different diseases, especially in the cardiovascular disease, as you are doing from bench side to bedside, in your setup, what are the biggest methodological or regulatory hurdles you have encountered in different trial design, especially if we talk about endpoints, durability of the effect, and also the immunological safety issues? So your take on that, sir.
Right. So let me take your last point. I mean, there's no immunological response because we are essentially taking the blood, getting the mononuclear cells, and examining the cells from there on, right? The progenitor cells. And as I mentioned that the only way to get to any form of information about endothelial health is to get the CD34 positive cells from the peripheral blood or CD34 VEGF receptor 2 positive cells from the blood. The progenitor cells, as I said, carries a lot of information. I didn't go into gene expression studies, but that gives a lot of information about how the cell has responded in a short period of time. And you would be amazed that the stem cell genes change within 2 weeks. We have actually showed that if one exercises, enos increases within 2 weeks. And similarly, if you don't, it takes a little bit longer. By 6 weeks, it will fall back. So the supplyability of stem cells for future is actually amazing. It responds way faster than any mature cell out there in the human body. So one can regulate and one can follow a disease process much more intently and much more intimately as the process progresses. Your other concern about so-called regulatory hurdles, well, the only intervention we are doing is whatever the patient is taking. But the question is, what responds for you may not respond to another person who is from Hispanic origin or somebody from Northern India versus Southern India. We know that our genetic makeups are similar, but not same. One might respond to SGLT2 or DPP4 quite fantastically, but another part of India with other genetic makeup may not. So how does one get to individualized medicine? I think the cell-based assays are much more robust, gives more individualized access, and not just serum-based, which changes from day to day. And as far as regulatory hurdles, I haven't actually seen any regulatory hurdles. We try to keep our processes quite consistently across the various trials. We have, as I said, we have done DPP4, SGLT2, GLP-1, exercise, heart failure patients, and we have amassed a huge amount of data. And as I said, the most consistent result is beyond the CD34 number and gene expression is the migration study. Migration in stem cell or endothelial circulatory cell migration is a major problem, and that's why the blood vessels don't heal and the perfusion does not happen. Even if you transfuse endothelial progenitor cells in a diabetes, it hasn't worked until now because you're putting in good cells in a bad environment.
Yeah. So Thank you, sir. So one question that has been asked by Dr. Mousumi De. What are the effect of progenitor cells in the cardiovascular medicines or therapeutics?
Right. So as I said, we have not yet figured out, well, there are a lot of emphasis on why the stem cells have not moved from bench to bedside. And the prime in cardiovascular realm or cardiovascular disease realm, and the prime reason is you're putting in stem cells which has bona fide regenerative property in a diseased environment. And they don't simply survive, and they undergo modification quite dramatically as soon as you put it in a toxic ROS mediated environment. So one needs to give it some protection. We boost up the antioxidant therapy. We put in various SOD2 or catalase, upregulate them using gene therapy. And then we have transplanted, and it has shown dramatic effect. Similarly, we have used P53 modification transiently, which gives some protection to the stem cells before you transfuse them. And these two therapies seems to have worked, and that work has been funded by American Heart for the last 10 years now in our laboratory. So with modification, stem cells has a role. Without modification, they have very little role in a disease process like diabetes. Even if you control the disease process, you can say, oh, I'm going to reduce his A1C from 10 to 7 and then transfuse. Doesn't work.
Yeah. So thank you. Thank you. We will take the last question from one physician member from API. He had mentioned like that, given the promise of endothelial stem cell therapies in improving vascular function, what clinical markers or imaging modalities would you recommend for tracking early benefit in patients with diabetic or atherosclerotic cardiovascular disease?
Excellent question. So as I was trying to allude to, that the serum-based studies or imaging-based studies takes time. First of all, serum-based studies are very finicky. It changes, and there's one inflammatory molecule level today, another level tomorrow. So it doesn't really give a very consistent result. Neither does it give a comparative result between one group of patients to another group. Whereas stem cells, as I was trying to say, is the meat of it, right? You're trying to discern endothelial dysfunction using everything else except the endothelium, which is crazy. Where is the endothelium? We talk about endothelial dysfunction. Was any of those studies to do with cells, or was it looking at something else? It was looking at endothelial nitric oxide dilatation. But again, if you have to follow a disease, you should look at the cells rather than the serum. It's like trying to diagnose and follow let's say, thyroid cancer without a cell diagnosis. I don't think any endocrinologist would ever believe a BRAF mediated cell, so-called serum, without any cells. Now, as you know, the entire BRAF diagnosis of thyroid cancer is based on cells. So why doesn't we do cell-mediated diagnosis in diabetic cardiovascular disease? So I think that gives a very consistent, individualized response to a therapy, whether it's GLP. Now in the realm of GLP-1, it's even more critical. There's more studies coming out about long-term side effects of neuritis and so on and so forth. One needs to know who is it going to help and who is it going to harm. How do you know? This is probably the most non-invasive way of getting to the cells.
So thank you once again. Thank you very much for your all explanations. And I think we have Professor Proshun Chatterjee with us. Professor Proshun Chatterjee is a senior professor at the Department of Geriatric Medicine at AIIMS, and he's a very renowned clinician, researcher, from AIMS. May I ask Professor Prashun Chatterjee, sir, please unmute yourself if you want to have a comment on.
Oh, thank you. No, I'm sorry. I joined late. As usual in AIMS, it's always in clinic. Clinic to basic is little difficult, but always in clinic. No, I heard few last opinion of Dr. Subraj. I think he's doing a great job. I just mentioned about I'm also working with mesenchymal therapy for frailty and dementia. So yeah, I'd love to get connected with you. I'm also sifting for the same work with from AIMS to some other hospital for this purpose only, because I'm keen to explore the possibility as much we can, because frailty, dementia, sarcopenia are something we just say it is like age-related changes, nothing much can be done. That is the worst answer as a doctor we give to patient. What we don't understand, we say it is age-related changes.
Yes.
So so there I have lot of interest, and fortunately I'm getting good researcher to work with, and we have a good dedicated lab where we are planning to extract all this and work on it. Many disease has been explored, obviously, just like the thing you mentioned about heart failure in pressure sore and other things, but frailty, one trial of course has shown phase 2 trial has shown a lot of promise, Kratos trial I think. And then so we are working in the same model, and I'm also trying to connect with some condition media if we can improve the quality of cell, quality and quantity of cell. So
Yes.
I think today there are other speaker also, but so I'll not take much time from it. But if I get your mail ID, I can get connected with you and get your guidance.
Sure. It's SSEN1 at
a second, just
gwu.edu.
I think
I can
Sambo,
share.
you must be having.
I shall share that with you.
So I took a permission from you. Now I can take your mail ID and I can mail you, and I'll love to have and I think it is great that less participants, so there are a lot of interaction.
No, not less participants. We have more than 500
Oh,
delegates
okay.
with us. So
This is for
the faculty link only.
7 faculty.
So we have almost 526 participants who are watching us. So once again, we are really thankful to Professor Shen, because your lecture was so important for all of us, because in India, the stem cell research is not growing that much. We have various regulatory hurdles. We'll discuss in our panel discussion, maybe on that. We have Dr. Nandini Kumar, who is an ICMR, ex-ICMR DG. So thank you again, Dr. Shen. Now we are moving towards our next scientific.
I'll sign off if that's okay.
Yeah. Thank you, sir. Thank you.
Yeah.
Good day.
Thank you.
So now we are moving towards our next scientific session, and that will be on geriatric research. And I think we had a small discussion how the geriatric population, they are called as clinical trial orphan. And we have a very renowned speaker with us today, Professor Prashun Chatterjee, who is a distinguished clinician and clinical researcher in the field of geriatric medicine. Currently, Professor Chatterjee is attached with AIMS New Delhi in the Department of Geriatric Medicine. He will tell us how to design inclusive clinical trials, how we can include the neglected population like older adults, and what are the issues regarding innovations, ethics, and evidence generation. Over to Professor Prashun Chatterjee, sir.
Thank you, Dr. Sambo, and I feel you are doing a great job by connecting a lot of people through this virtual media. Nowadays, meeting face to face has become very less after COVID crisis, right? And it saves a lot of time for us. Like my slide is visible to all?
Yes. Please make it full screen. Yeah.
So thanks for the opportunity. And first of all, I'd like to say I'm not a great expert of clinical trial for older adult, but yes, what I understand is their problem. for my last 2 decades of experience of managing senior citizen. And many times we feel orphan when we cannot say that we say that we don't have any evidence to do this. So that fine barrier is always very difficult to explain to the patient many times. So it's more of my, as I mentioned, it's more of my experience. And I tell this as ethical blind spot and of not recruiting older adult in usual clinical trial, just like the way we miss many things due to our blind spot. So older adults are often excluded or poor represented in clinical research. It's more than scientific gap. I will say it's more of ethical blind spot. So the problem of exclusion is we often consciously excluded individual with frailty, multimorbidity. Frailty is a cumulative deficit model where people, due to your lifelong deficiency in some of other organ, mostly physiological function get reduced, and we have some phenotypic criteria, but still frailty has not get a score in ICD because of poor model of definition. Similarly, multimorbidity, as you know, multimorbidity is more of rule than exception. So again, same person has hypertension, diabetes, coronary artery disease. Along with them, they had a stroke, they have osteoarthritis knee, and other problem together. Along with that, functional decline or dementia from clinical trials, treating this condition as inevitable consequences of aging. The things what I was discussing, when we don't understand, we say this is a age-related changes, nothing much can be done. Please accept this as age-related changes. So that is a major problem, I believe, of exclusion. And there has been limited treatment, partly due to lack of motivation from both patient as well as from the medical fraternity. Because I believe most of this condition can be treated, and we try to give them some or other treatment, be it pharmacological, be it non-pharmacological. As a result, many key geriatric syndrome remain underrepresented in clinical research, and maybe there is fear of adverse event, which is true, because even frailty definition also we say any small insult, the person's functional decline will fall drastically. So we have that fear of adverse event for any clinical trial. Polypharmacy, cognitive impairment, regulatory hurdle, which is recently discussed, and sometimes pure ageism. Recently I sent a patient to a super specialty unit for intervention, cardiological intervention. He has been clearly denied treatment because he is 95 years, even though his functional status was absolutely perfect. And that I gave a note still. So this is pure ageism. Then how can you create evidence when you are not interested to intervene, right? This leads to a situation where evidence we use to treat older is often based on trial that never included them. So it is again a subjective treatment we start in this population. So ethical imperative, we must remember respect for participant, beneficence, and justice. So respect, demand, informed consent process adapted to cognitive or sensory impairment, because we have to understand regarding cognitive impairment, it is like from 60 to 100, it increases exponentially from 0.9 to 7% in 60s to 40% for centenarian. Now we have good number of centenarian also in this country. Then along with that, there is hearing problem, there is vision problem. So still they demand respect, and the amount of time you have to spend to get the consent needs consideration. Same beneficence job means generating evidence that actually helps them to live better. It's not always only theory which is going to help you, or for our learning, it has to give them a better life. Justice requires fair inclusion and should not be at the burden of disease without access to benefit of research. So exclusion is not protection, it is a neglect. So inclusion does not mean lowering scientific standards. It means innovating the way we do research. This is absolutely true for geriatric population, especially someone who has multiple health issues together. So I feel pragmatic trials are known to everyone. it allow inclusion real world older adults with more comorbidity, real life settings, improve external validity and applicability. Similarly, we have to adopt few new things. We have to be flexible, interim modification, making trial more responsive to the heterogeneity of older adults. Because one thing here I have to underline that heterogeneity is too much in older adults as compared to any other population. Because of these various hallmarks of aging, various changes, more you age, more you become heterogeneous. So there is another added challenge which we have to address. Similarly, geriatric assessment tool like CGA, ICO at baseline can stratify risk and personalize outcome. We have to decentralize our trial model, either from hospital, even from the community. Maybe we can use telemedicine, remote monitoring, home visit by the research team for frail homebound elders, instead of considering that excluding them. So we have to redefine outcome. Older adults, they don't care about their survival. When they come to me, they say, beta, it's like, I have lived my life. Now you try whatever you can do. But one thing, make sure that my quality of life should be intact. My day-to-day activity, I should able to go to loo. I should switch off my TV. I can take care of my finance. I should not depend for that. So please work on that, focus on that when you are designing something about outcome, because till that the outcome is primarily focused on biomarker mortality, but much more important is their function, independence, quality of life, activity of daily living, gait speed, mood, loneliness, caregiver burden, patient reported outcome is equally important. Even nowadays, studies are showing subjective feeling of aging can also be an outcome, and that makes a lot of sense. Studies shown that centenarian has better subjective aging feeling. Subjective aging means they feel they are younger, so they live long. So then informed consent also needs some innovation, because they may face hearing, visual impairment, cognitive decline, digital illiteracy. So we have to work on that domain, how to get proper informed consent. We must use simplified language. We are very busy in every space, including in research, and we try to bypass many things. So that is difficult for elderly. We need audiovisual aid. We need re-consent procedures sometimes too. Many times they don't understand, and single shot you have to give them more time to make them understand. It has to be legally acceptable representative when needed. So inclusion also means rethinking informed consent. Of course, regulatory push, global momentum is there. Now FDA, EMA now require age subgroup data. Landscape is definitely changing. WHO has declared this decade as decade of healthy aging. So WHO is also pushing more geriatric inclusive research. ICMR guideline now emphasize on vulnerable population, yet we need more implementation at trial registration and ethics committee level. It's still difficult. I also many times I didn't get approval to do even calorie restriction trial for elderly, giving metformin. It needs a lot of hurdle to pass. And every trial protocol should justify exclusion of older, not inclusion. So yeah, and this is an urgent call, because we are entering an aging society by 2050. All of us know more than 20% older adults. So we will, like, older population and pediatric population will be same. The decision we make today in clinical trial design will shape the future geriatric care. So at least we should create evidence for 2050, and we have some time for that. We need evidence of polypharmacy intervention. We need pharmacological, non-pharmacological therapy for frailty. Just I was discussing, we need dementia treatment, not only prevention. We need to reverse dementia. We need longevity science, which is my primary area now, and regenerative medicine in real world elders. And I feel this is possible to generate that much evidence when we will do personalized medicine for each and everyone, irrespective of their pocket, and for all. And that is also possible. So yeah, so let's generate evidence for them and with them. So like one trial which I have written now, and I'm working on this sarcopenia treatment, because till that we generically we say exercise, protein supplementation, but let's say emerging therapy is NAD booster, and older adults with frailty or cognitive impairment are often excluded, but they are the one who need it most. That is the fallacy we have to address. yeah, so we need to inclusive, pragmatic, ethical, and outcome oriented. So inclusive trial are not just about better science, they are about equity. I would like to stress here because being a proponent of geriatric medicine, I'm a strong proponent of elderly equity, their dignity, they should speak about them. They should decide about research. Even the person has to be part of the research team. That is happening in many places. Then as a clinician, researcher, policymaker, we owe it to this generation, and we build a healthcare system we work in to ensure they benefit from its progress. So more of tokenism to transformation, from protectionism to partnership. We don't only, they don't want sympathy from us. They want more evidence from equity. Yeah. So thank you. I'm happy to take any question.
Thank you very much, Professor Chatterjee. I think this is very much important for all of us to understand how should we become inclusive to this clinical trial orphan population when we are talking about geriatric population. And I think one issue I just want to ask you, that even we talk about the elderly population, in that population also there are lots of heterogeneity. I think there could be some individuals who are independent old people, maybe 70 or 75 years old, without any disease or with any disease. There could be some independent but having some multimorbidity, or some frail older people, but they are independent. And again, now there could be some population who could be complex, multimorbid older patients with disability, certain level of cognitive decline. And now another group that could be living in some institutional care, multimorbid older patient. So when we talk about clinical trial inclusivity, how we should approach in this each group of patient, because all of them requires inclusiveness in the clinical trials. What is your take on that?
Absolutely, absolutely, you are right. So that is the thing we are mentioning here, that the point is, with aging, your heterogeneity increases exponentially. It's not only because of your social status, not only because of your multi-morbid condition, but also so now scientists has found out there are 12 hallmarks of aging, including DNA instability, mitochondrial dysfunction, then epigenetic alteration. So epigenetic alteration can happen. Let's say someone is under stress with all same profile, clinical profile. Someone is not under stress. That also changes epigenetic profiling. So we have now enough explanation to say why heterogeneity is increasing amongst elderly people. So if you ask me what is the best way to do this, because obviously we are not in the ideal world, best way would be to assess biological aging of individual and recruit them in the study, which we are planning. So if I get, and we have to now understand every organ is aging differently. So heterogeneity within an individual is also there. So the way, let's say someone who is at the age of 80, the way, or even at my age, at the age of 46, my every organ is not aging in same speed. So we have to have our cellular aging, what we call as biological aging. So more pragmatic approach would be to go for, if we are not going for precision medicine, for layperson language, if I say, your functional capacity should be the criteria which will define an elderly person, which will cut short many of the other heterogenic processes, including financial status, socioeconomic status, educational status, etc.
Very important. I think this is a very revolutionary comment from your side. We have to move from age to biological age rather than age is not a number.
Yeah.
We need to first consider that. And straight away in all our clinical trials, we are mentioning from 18 to 65 years, we are including them. So this is I think this is a very important thought-provoking statement from you, how we should give emphasis on the biological aging, and regarding with respect to biological aging, we should include our elderly population also in different categories. Another important issues when we are talking about trials in elderly population, we know that there is a possibility of multimorbidity, and patient will take multiple other drugs for different comorbid conditions. Now, generally what happen in traditional clinical trials, we exclude those patients who are suffering from multimorbidity. So here is again a drawback for the elderly population. And I think we need to become more pragmatic. And when we are doing research design, especially in this group of people, we should follow the ethos of pragmatic clinical trials, the inclusiveness. So what is your take on that?
Absolutely. Actually, it is very rare to get a 80+% without multimorbidity. Multimorbid means two or more than that. So hypertension, diabetes, ONE is so common, right? So now WHO has come up with a concept which I was also fortunately the part of it. We can assess their intrinsic capacity. Intrinsic capacity is it includes your vision, your hearing, your mood, your cognition, your mobility, your nutrition, like that. So once we have, again, the coming back to it, is one form of holistic assessment of your functional capacity. So what matters when we are following a person is their functional capacity, be it whatever may be the reason. Even the point I was mentioning, frailty in elderly, is also a overall functional decline. So functional capacity is, I can say, one of the phenotypic expression of the all 12 hallmark of aging, what we are covering. So hallmark is in cellular level, intrinsic capacity is your phenotypic level. So definitely aapko agar, if you don't get anybody who has no multimorbidity, then will you not do trial on 85 or 60? Even the trial now is going on centenarian. So people are now thinking there is a concept called super centenarian. That means whose brain function is still good. So cognitively, they are still intact. So only one criteria we need to follow in centenarian trial is that their brain function should be okay, so that we can follow them. Their rest of the sensory can be taken care.
Thank you, Professor Chatterjee. So again, you had re-emphasized the need of pragmatic clinical trial, and especially when we talk about clinical trials, we always think about the internal validity, but I think we need to increase, we need to check how we can increase the external validity of those trials. So thank you once again. And I think all of our delegates had been there, and they were being very much benefited by your lucid and informative lecture. So we are now moving towards our next session and the final session, and that is one panel discussion we have, and we will start panel discussion within 2 minutes of time. I request all the delegates, please be with us. We'll start our panel discussion by another 2 minutes.
Thank you, Shambo. Thank you all. Thank you for calling me here.
Good evening, everyone. I think we are a little late and very sorry for these technical discrepancies. So we are really delighted to have with us our respected panelist. And the topic of today's panel discussion is the future of clinical trials in India, integrating innovation, inclusivity, and ethical rigors. So we have with us Professor Santanu Kumar Tripathi. He is the president of Association of Clinical Pharmacologists and ex-head at the Department of Clinical Pharmacology, School of Tropical Medicine, Kolkata. We have with us Dr. Nandini Kumar. She is president of FERCI and also ex-deputy director of General Senior Grade at ICMR. We are also delighted to have with us Dr. Srinivasa Murthy L. He is also a very senior physician and clinical researcher. He is the vice president of RSSDI, and currently he is attached with Life Care Clinic and Research Center at Bangalore. We have with us Dr. Santosh Taur, Director of Medical Affairs, Vaccines and Digital at Pfizer Limited, Mumbai. We also have with us Mrs. Priyanka Bosch Bhattacharya, Head at the Department of Medical and Clinical Governance and Process at Abbott Healthcare Private Limited. We also have with us Dr. Anirban Rai Chaudhary, Associate Vice President and Head at Development Center of Excellence, Sun Pharma. On behalf of PRF West Bengal branch and API West Bengal branch, FERCI, IPS, and Association of Clinical Pharmacologists, I welcome you all in this panel discussion. So I had shared one slides with Clarnet team. May I request the organizer to please share the screen for our delegates who are attending today. I think almost now it is showing around 540, 542, like that. Really thankful to all of you who are there from 2 p.m. today. Today, we will try to cover this broad agenda. Number one, how we can implement innovations in clinical trials. It could be artificial intelligence, medical devices. What should be our inclusive research design, especially when we talk about underrepresented societies like older adults, rural people, tribal people, women, vulnerable population. What are the different ethical rigor and evolving regulations in Indian clinical research? Operationalizing recent CRO registration regulations and their implications, and overall role of sponsors, regulators, and clinicians in sustaining participant trust. So, as there is, I'm sorry the slides are not being shown, but may I go through the important landmark? As already we had discussed, the International Clinical Trial Day.
Excuse me, sir.
Yeah.
Hello. Sorry for disturbing, sir. Sir, just give me
You
2
got
minutes.
my
Yes, sir. I'm sharing the screen. Just give me 2 minutes.
But Shambo, you can go on, I think. You can go on meanwhile.
So I think we can go on. So we know about the story of HMS Salisbury, the 12 sick sailors who were divided into 6 pairs. Then ultimately what we got, that 2 oranges and lemon, that works. And from 1747, that results ultimately translated into their regulations. 1795, British made citrus compulsory in the diet of sailors. Now, when we talk about few important achievement by which after this Lind's treatise of the scurvy we got, first of all, all subjects were in similar condition. So like was compared with like. We know the importance of controlling the variables. But as we are talking about from 1747 to 1795, long time it took. Why? Because they thought they will make that perishability of the fruit and expense, they make it inspissated juice. So ultimately what happened? The vitamin C was not there. So there was some confusion. And I think with all clinical trials, we have certain confusions. And for that, we need to find out the answers. We need to do further research. And there were previous documentation that was being missed. Maybe during the Vasco da Gama and Hawkins, they had mentioned about the importance of oranges and lemons. So again, it re-emphasized the review of literature, previous documentation, which matters. And after this important study, another area we had explored, how to distill fresh water from seawater. So again, that had emphasized the importance of occupational health, and we got something called as secondary outcomes from a research. So in that perspective, I think the ICTD is something very important. You can move forward to the next slides, please. We had already introduced our respected panelists. Next, these are the themes. Next. Yeah, this is the famous book, The Treatise of Scurvy. Next. And we had discussed this ICTD. For that, we are celebrating 20th of May. Yes, next, please. Next. I think this slide is very important for those who are postgraduate students, because often we use this terminology, clinical trial and observational studies together. First of all, we need to understand as investigator whether I am assigning the exposures. If the answer is yes, it is experimental study. And that is randomized control trial or non-randomized control trials. So if we talk about clinical trials, then we need to decide whether as an investigator, I am assigning the exposure. If it is not, then it is an observational study. This is a very simple rule. If as an investigator, I am not assigning the exposure to the patient or participant, then that is an observational study. Next slide, please. Next. So if we talk about the experimental studies or the clinical trials in India, we need to have a thorough knowledge about this document, that is New Drug Clinical Trial Rules 2019, and there are certain updates. Next, when we talk about observational studies, this ICMR guideline is there to help us. There are certain overlap between these two documents that we can discuss. Next slide, please. And this should be the first statement we need to understand when we talk about different key stakeholders in clinical trials. We do research for the pupil, and that should be with the pupil. So there should not be on the pupil like behavior. Next, please. We have with us now Dr. Nandini Kumar, who is a renowned ethicist attached with multiple ethics committees, and currently Madam is President of FERCI. Madam, I have two questions for you. First, with increasing inclusion of vulnerable populations, it could be terminally ill patient or cognitively impaired patient, how do you see ethics committee navigating proxy consent and distributive justice, especially in community-based studies?
So before beginning, I once again want to thank you for including me in this panel. In fact, in the ICMR guidelines of 2017, we have a section on vulnerable group. And when this was actually being designed by the committee for that particular section, there were 3 A4 size papers where these vulnerable groups were actually described. But you can't use that list in the ICMR guidelines. So with great difficulty, actually, I condensed it, trying to see that I do not leave any any vulnerable group as such and shortened it and saw that it was inclusive of all types of vulnerable population that we have. In fact, I always say that any research participant for that matter is vulnerable. It's only about the relativity. One may be less vulnerable than another. So, for example, in in a cancer institution, stage 1, a person suffering from stage 1 will be less vulnerable compared to a patient suffering from stage 4. So it's a relativity which is very, very important. And the ethics committee's duty is to see that their interests are protected. How can you actually protect that? The most important aspect is the informed consent. Most of them will not be either, their situation would be such that they will not be free enough to say no. And this situation could be varying for various reasons. They may be, age-wise, children, they are dependent on their parents. And older people, they are dependent on their children. And we heard a lot about the geriatric population in the previous talk. And it reflects a lot on me also because I have also entered that group now. And I very much feel that if there is a trial where I can be included, I will very gladly be part of that. But if you actually consider my vulnerability, I'm not as vulnerable as another person who may not have such knowledge or awareness or may be dependent on his household, other mates or circumstances. Think about the institutionalized inmates, whether they are children or widows. We have such a facility in our country for widows. And also old, the senior groups in, being left in institutions to fend for themselves. They're all, people who cannot give their consent, like wholly understanding the situation. So who will actually speak for them or be responsible for them? And they are called actually legally authorized or legally acceptable representatives. That's the term we have put in the guidelines, because in some countries they talk about legally acceptable representative. But when we talk about legally authorized representative, it means the parents or the legal custodian. So when a disabled person is there who cannot understand the nuances of whatever you are talking, another person has to come and help. And that person could be anybody, parents or legal custodian. If not, could be grandfather, uncle, aunt, or caretaker. And when you are talking about institutionalized people, then it will be the head of the institution responsible for that. So they have to take care of that. And two things that we have to consider when talking about vulnerable groups is include them only if that research addresses their problem. Or the other side is, if a vulnerable person satisfies the inclusion criteria, that person has every right to be included in the study. Just imagine, say, a 16-year-old child in the school suffering from melanoma, coming from a lower socioeconomic group. Now, if there is a trial site somewhere near where she can actually go to that unit and get treatment, test, drug, is it not okay if she goes there? Would you actually try to say that, oh, you come from a lower socioeconomic group, we don't want to include you because tomorrow people will question us? That's not the way things have to be seen. one has to see whether they actually satisfy the inclusion criteria, and that you are not forcing them to enroll in the study. There'll be other forces. So the ethics committee will actually watch out for that. Another way of protecting them would be, ethics committee would say that there has to be monitoring. And that monitoring can be off-site or on-site. Off-site means you get the progress reports, etc., etc. And especially if there is more risk, you will ask for progress reports at a shorter interval instead of waiting for 1 year. Generally, we wait for 1 year. So you'll ask for 3 monthly or 6 monthly. And during COVID times, there were even requests for 1 weekly report. So it depends on the type of risk that they would be subjected to. So that is the monitoring. And also you can take an active role. You means the ethics committee can take an active role in visiting the site. Suppose this is an institution, then you can go and see for yourself how things are. So, and if it's a community, you can visit that community. And this community doesn't mean that it's a group in a village. Community can mean anything. It's, for example, a community having similar disease, that is also a community. So community is a very broad-based term. So that's my take on the protection to be offered to the vulnerable groups by the ethics committees.
Thank you, madam. So you tell us how ethics committee should become a more proactive rather than only reactive. So now moving towards the next part of the question, next question actually. If we talk about ICMR guideline and their implications on different Indian ethics committees, now we are talking about artificial intelligence era. So how ethics committees evolve to evaluate multicentric artificial intelligence led trials where explainability of risk is limited?
Actually, a very complicated question as such, because multicentric trial itself is a topic by itself. And when you add AI to that, it becomes even more complicated. First of all, when you have multicentric trials, let's just simplify that first. When you have multicentric trial within our country, then one needs to actually identify one particular ethics committee as the designated ethics committee. This actually is not in the AI guidelines of ICMR, but it is there in the 2017 guidelines. So then you have a separate guideline also on multicentric trials by ICMR. So the point is that this designated ethics committee will take inputs from all the site ethics committees and make the master plan. And once the thing is finalized with the inputs, then it is actually sent to all the site ECs. Now it is up to the site ECs to either accept it as such, or technically they want to actually have more rigorous control. Then they can say that, okay, in their SOP, if they describe that if this such a thing comes, it will go through expedited review. So in the expedited review SOP, you will have to add that. Now that is about multicentric trials. Now when it comes to AI, it is slightly different in the sense about the expertise in the ethics committee. So not all of us are, AI trained or anything like that. So even I used to actually avoid, learning more about AI. I used to run away from that, but there's no other go. You have to face it. And I've started facing it. So what the ICMR guidelines say is that in the ethics committee, we have the legal expert. But you have to choose a legal expert who is conversant with IT and also has done medical law. How medical law is going to help in AI, I don't know. Medical law as such would be an additional qualification. And then you have to have data scientists in the group. You have to have computer scientists with AI knowledge. So subject experts, of course. So either it's not written co-opt, but you can co-opt. But our guidelines as well as the regulations of the country say don't go beyond 15, number 15, 7 to 15. So you adjust that way, have these experts. That's the specialty about the EC structure. Now, not everyone can understand AI, so you will have to give them some training. This is only for those ECs which is actually going to review the AI trials. So, I mean, AI associated, I would say, trials. So this training should be in big data, Internet of Things, and also about how data collection procedures should be. It should also be told about the importance of getting progress reports, monitoring. The type of review that will happen will be the same as for the other types of research that we talk of, like exemption, expedited, and full committee review. That depends on the type of risk that is involved in the study. So there are many other things. Main thing is that, when you are collaborating in a multicentric trial, you have to be very careful that you are all on equal footing. I mean, the centers are all on equal footing except for the designated EC, which has more responsibility, overall responsibility. But the site ECs will be responsible for their site conducting the trial. Conflict of interest is very, very important in that. Biological and data transfer has to take care of data sharing, data security, and whatever regulations are there. In our country, actually, there are no specific regulations as such, but the Niti Aayog also brought out, just let me see that. Well, I'm not getting that, where it has gone.
I think we have now the DPDP Act also.
DPDP is there, but yeah. The Niti Aayog actually has brought out principles of AIs, like safety, inclusivity, privacy, and accountability. Then we also have National Digital Health Mission talking about reliability and safety, data handling, patient consent, valid data, and the use of AI tools. This is very important because the AI tools that you are using should come from a very authentic source. Earlier, we heard about, the manufacturers who are not even registered or who don't get the CDSCO approval actually offer to have the diagnostic tests done. Similarly, about these AI trials, you have the supervised, semi-supervised, and non-supervised, adaptations. So you have to actually see whether these are supervised ones. And that means some sort of standardized methodology would have been used. The data collection, what data got fed into creating that algorithm, that is very, very important, because otherwise it can lead to false data. And who do you include it has to be, you cannot exclude certain groups. So you will not get a proper conclusion because of that. And not only that, when you are using AI, the tools can malfunction. So you need to have a lot of monitoring from that point of view, quality control of that. The ICMR guidelines talks about 10 things that have to be actually seen. That is about autonomy, that of course that brings in consent, safety and risk minimization, trustworthiness, data privacy, accountability and liability, optimization of data quality, accessibility, equity and inclusiveness, collaboration, non-discrimination, validity are the 10 points. I will not elaborate on each point because it will go beyond time. So for the time being, I think probably.
Yeah. No, thank you. Thank you, madam. I think one of the very key take-home message, whatever you had tell us from that would be artificial intelligence may be borderless, but ethical accountability is not. So ethics committees must upgrade. We are now protocol reviewers only, but also we have some capability now from the ethics committee perspective that we could be an algorithm. There are different algorithms we know. So algorithm interpreters and data protection, data privacy and that protectors also. So I think we should have artificial intelligence specialist in our ethics committee. That is what Madam emphasized. Now, please go to the next slide. Yeah, so we have with us a renowned clinician of this country, and he is a very reputed clinical researcher, Dr. Srinivasa Murthy L from Bangalore. So I have two questions again for you, sir. So number one, from your clinical trial experience, how do we balance clinical judgment with protocol constraints when trial participants present with different comorbidities? For an example, diabetes, cardiovascular disease patients, or it could be infections also.
Thank you, Shambo. I'm literally honored to be present among so many stalwarts in the field of clinical trials, and happy Clinical Trials Day to everybody. And Shambu's question is very apt because in many cases, the protocol does not define what do you do when you have a certain condition. So it falls down to investigators' discretion. So very commonly, the problem is shifted to investigators' shoulders. That is where we end up in trouble, and that is the time we need to use our clinical acumen. I think that is something which most of us lack in our routine clinical practice, that forgetting about the clinical acumen and correlating, because end of the day, it is the patient's safety what we should look at. And that we tend to compromise. For example, there can be a deviation in one of the trials. The NT-proBNP was to be done only when there is a suspected case of heart failure. And the tubes containing the lab kits add a tube for NT-proBNP by default. So all the centers started performing NT-proBNP without checking for the heart failure signs and symptoms. So that becomes a protocol deviation. And this happened across the sites. So the problem now is that we have all taken protocol deviation, informed the ethics committees, but how do we go about because some of the patients in that subjects in that study had an NT-proBNP raised. So the PI went on to do a echocardiogram, proved that, and some of them they've treated with an anti-failure medicine. Now the dilemma is whether it was right or wrong. As per protocol, it was absolutely wrong because we used the tubes which was not meant to be used for that visit until unless there was a signs and symptoms of heart failure. So this is the confusion we face. So, but the PI's argument was that I have taken care of the safety of the patient. Yes, it is a mistake, deviation. Patient was asymptomatic, but the NT-proBNP was very high. So that is where we lie. So that thin line, there is always a gray zone end of the day, as PIs, it's our responsibility to take care of our patients' health. So we should not hesitate to deviate protocols at certain times. And of course, keeping the ethics committee in the loop always, and keeping the sponsor or the people who are responsible for that. So it's very important for us to take a call. And a lot of times, for example, right now we're doing a Rethinking trial, which is a triple G, lot of diarrheas are seen. So I had to go back and read the investigator's brochure to safeguard my patients for the patient retention. Because imagine a person having 4 or 5 diarrheas per day may not continue in the study, right? So I need to look at that as a responsibility as a PI. I need to look at the safety of the patient and how often all these happen, because that is a new molecule for me. I have not used it in my clinical practice. First time I am also getting exposed to the molecule. So how do you tackle that? How do you retain the patient? So the comorbidities, the patient's condition, clinical acumen is very, very important. The safety of the patient, as any person says, like in the FDA, the first thing they look at is the safety of the patient. For example, one of the FDA inspections I faced, the primary reason why they came into was there was a blinded CGM, which was double blinded, not to the patient, not to the investigator. So they released the CGM data post closure of the study. Okay, so they were saying it is to avoid the bias, which even the inspector was not very convinced. But once they released, we know patient had about 50-60 hypoglycemias, which was not picked up by your glucometer. So that was one of the signaling why they came for inspection to the site. That was an approval trial for long-acting insulin, and that is how they pick up signals. So it is very important that we report. And of course, now in India, of course, we have definitely upgraded ourselves. We are very upfront. We are through the world that we are doing a lot of ethical business now, and there is no unethical business in our country. And especially the ethical committees have become more stringent, more proactive. In fact, I wanted to ask Madam about the virtual meetings of ethics committee should be approved. Because physical meeting, post-COVID especially, we need to have that. So we have put it in the SOP that saying that some of the meetings can be virtual. It is not that every meeting can be virtual. Some of the meetings they have to allow, and they put it in the EC SOP. So the ethical committees, when there is a minor amendments, can meet up. So that is where the AI comes into picture in the clinical trial. And one of the suggestions I made in one of the insulin titration, there is a basal bolus versus a long-acting insulin comparator. In the basal bolus, there are a lot of hypoglycemia seen, and we don't get a pop-up. So every day we need to titrate 5-6 patients in the iPad, and the patients are given a Bluetooth enabled and with a SIM card, the glucometer. So as soon as they check, it comes to our iPad. But the iPad I'm not keeping with me 24/7. So when somebody thinks that 7 p.m. or 8 p.m. when I'm in OPD, I may not see that. So one of the artificial things that the AI part, what I was recommending, and which is being implemented by one of the sponsors, is that it should give me a SMS kind of a sound, or a WhatsApp kind of a sound that there is something wrong. So I go to the iPad and see. So that is where I think we should use this artificial intelligence, all that, to have a safeguard of the patient, to increase the safety of the patient, our subjects. And LAR rightly said by Madam that the LARs are the most vulnerable population, which are used or misused by many of the PA, which is a saddening part. Because, for example, a vegetable vendor on the roadside is parents, he cannot manage. So if you say a free drug, and I have heard in one of the forums that one of the doctors says, like the cancer medicine is like a boon, some few lakhs you are getting free, so you enroll. That is not the way we are supposed to do that, ethically, right? So these are some of the gray zones and some blunt mistakes people tend to do for retention's sake. That is bad.
Absolutely. So as clinical researcher or clinical trialist, you are wearing the two hats. So two hat conflict is also an important area we need to address. And for that, I think if you also can share your real world experience where trial inclusion and exclusion criteria excluded a population you clinically thought should benefit, but due to that stringent inclusion and exclusion criteria, you have to remove that population. So how can this also be addressed?
So it is very difficult to address or sort it out, because when a new protocol comes, obviously when I'm seeing my OPD, I will be thinking about the inclusion exclusion criteria of a patient sitting in front of me. But again, I will assess his educational status. For example, in my SOP, there's no LAR. I don't take any uneducated in any of my trials because that bias itself goes up. Because see, the LAR issue is there has to be a witness there. And who is the witness again? Now, how do you define the witness? The witness also can be biased. If I take my nurse as my witness, obviously she's my employee or my receptionist or somebody sitting outside is my patient. So I can ask him, he's a neutral person. But again, there I get biased. So that should not happen. So that is why I have made it a SOP that there is no uneducated or LAR used in my consent. And it's all AV consent now for phase 2, phase 3 as we know. So when patient is sitting in front of me, I would definitely think about the inclusion exclusion of a given study, whichever the recruitment is going on. So that is the stage where you tend to think, and it is not a bias, it is putting them upfront that there is an option other than your OPD treatment which is going on. If you are willing, I would like to explain. So many of patients, 80% of them say, yes, we are interested, they will listen. And 90% of them say no. That is the most difficult task. And thanks to our media and our other inverted commas friends who talk nonsense about clinical trials. I'm sorry to use those words, but they say you have been experimented, you're being exploited, all that, wherein it is an audiovisual consenting and they have to write, read, and then take a call. For example, in my OPD, I give them a consent form, to make a small note of it that so-and-so version has been handed over to the subject. If he's interested, I'll ask him to come back. If he's not interested, just return the ICF to the reception and again document such and such a patient has been returned. 90% of the cases. So I offer definitely when there is a, for example, there is a obese and comorbidities, there is a dual and triple agonist a trial going on. So I will offer them. There is nothing wrong because now, you know, the dual agonist is already in the market in India. So those are the, like tirzepatide I'm talking about, and we have done trials. This are bound to, we were part of, only 2 centers in India did that, and we were part of that trial. So that is where the question comes that wearing 2 hats is actually a blessing in disguise or advantages, I would say, because every in and day in and day out, you're doing the same. And you tend to identify subjects. And that's one of the reasons why I face the FDA inspections more frequently, because in most trials, I'm the global highest recruiter. So that is where one of the signals for the highest recruiters, you tend to face the FDA inspections repeatedly.
Thank you. Thank you, Dr. Srinivasa Murthy. I think we can conclude from your saying that balancing clinical intuition with scientific rigor, it is not a compromise. It is the cornerstone of ethical, inclusive, and impactful clinical trials. I think with that, may I move towards the next slide, please? Yeah, so we have with us a very renowned clinical pharmacologist, Dr. Santosh Taur. Currently, Dr. Taur is in the Pfizer as in the medical affairs and digital team. So we are really delighted to have you. So my first question to you, in a post-pandemic India, how is industry integrating digital health tools and decentralized trial models in alignment with Indian regulation?
Thanks, Shambo. I think this is quite insightful conversation. Am I coming through clearly?
Yes, yes.
Superb. So I think India has seen a significant revolution with regards to the conduct of the clinical trials. While we still hope that there could be a faster pace of the revolution also happening towards the regulatory framework, and that's also taking its own pace, and which is also in more the positive manner. I would like to highlight that while we all talk about decentralized clinical trials, and we know it is a complete spectrum. On extreme right-hand side, we have fully centralized clinical trial, which is our traditional mode of conducting clinical trial. And then we have extreme left-hand side, where we have fully decentralized clinical trial, where most of us haven't reached there. Okay, because in the current construct, we know that there are certain elements that still requires human touch. And that's the reason we are somewhere between the 2 spectra. Okay, that's called a hybrid. So we have certain components of the traditional clinical trial where the first visit happens at the site, and maybe certain subsequent monitoring visit may happen at the remote site or at the home care facility. So we are still, I think, hovering around this hybrid spectrum. And now I think there's steady movement towards the fully decentralized clinical trial. Now, what model is working? COVID has accelerated decentralization of clinical trials. However, it was not the period of genesis of decentralization clinical trial. I remember like Pfizer was the first to have investigational new drug approval for any clinical trial, which was approved way back in 2011, which was our remote study. It was research on electronic monitoring of overactive bladder treatment experiences, which was used for this condition. And then we had different components, right, from e-consent, e-monitoring. All these were first time used in the IND approved, FDA approved clinical trial design. And thereon, I think we have seen a lot of clinical trials, and there were many examples before also. And there were few companies who did trials with certain components of digital technology, which were used at different aspects of the patient journey. So what we have seen in the journey is that the decentralization of clinical trial can happen at multiple places. One biggest area is uh, the patients need to visit the site. Okay, and that will depend on complexity of procedure. Okay, there are certain procedures which are less complex, like your vital monitoring, your electrocardiograms, or sometimes there are less complex procedures such as injections. We don't have to visit the actual clinical trial site for that. And that's why we call something called as like how do we reach the last mile, wherein we use the local healthcare facilities. Like we have many community health centers. Okay, we have the hospitals. Okay, how can we use them? Second concept is the mobile health technology. Now we see a lot of mobile health reaching the patients and the participants home. So can we use those mobile health units, which are called as mobile research units? So you have the satellite centers in addition to your main clinical trial center. And third is reaching the last millimeter, not the mile, millimeter, where we have home-based nursing. Okay, home nursing in clinical trial, HNCT, where some of the services, including lab investigation, certain monitoring, can be conducted in the comfort of someone's home. Okay, that's about the clinical site related activity. Then the second most important component is about the assessment of the endpoints. And we have seen that EDID has been increasingly used. And we have clinical outcome assessment, which is further divided by various regulatory authorities into uh, PROs. And now we have the electronic counterpart for it, patient reported outcome, EPROs, EPROs. We also have PERF outcomes. Okay, we have clinician reported outcomes. We have observer reported outcome. And all these outcomes can be converted into electronic components. In fact, now we also see digital biomarkers. So there are certain biomarkers which have been digital markers which have been validated by the health authorities. And we have seen many examples of these for atrial fibrillation studies. There are many studies like Apple Heart Watch, Fitbit Heart Study, and now these have been approved by US FDA. Okay, and these biomarkers are increasingly used in the clinical trial. So definitely, I think we are seeing significant improvement there. And third important component which I want to highlight is the patient recruitment. And we heard recently Srinivasa talking about how the unethical practices may happen. And there are good data which show that conventional recruitment methodologies, they lead to delay in recruitment at 35% of investigational sites and more than 80% of trials exceeding their enrollment timeline. And now what we have seen when we apply different elements of decentralized clinical trial monitoring, the overall development can be brought down. And this is proven that in 2019, we saw that it used to take on an average 8.6 years. But in 2022, it came down to 4.8 years. So this shows that if you use multiple digital methodology, including digital and social media platform for patient recruitment, it will lead to much faster patient recruitment. And how does it culminate into? Everything that we do will lead to faster completion of clinical trial and eventually better and faster access to these innovative medicines, biologics to patients. Yeah.
Great. So I think what you had discussed just now, we we can understand the future of clinical trials in India will not be built in hospital corridors alone. It will be co-created through three C's. One, cloud platforms. Two, community homes. And three, connected devices, with regulation working in step, not behind. So with that, I want to ask you the second question here. In cases where digital innovations in clinical trials encountered regulatory hesitations or resistance, what proactive strategies, such as integrating real-world data or hybrid trial designs, can be employed to align innovation with regulatory expectations?
This is a very important consideration, and I must appreciate India did its part much before even pandemic. We had our EHR guidance released in 2016. We had our telemedicine guidance, which was released immediately after COVID pandemic in 2020. While that time it excluded clinical research, clinical trial, and we are hopeful that we would have certain components of telehealth, telemedicine, which will also address a clinical trial. Because that time the requirement was more to deliver healthcare to patient doorsteps. And that's the reason the regulatory guidance was more focused on the telemedicine part. Now we have to see with the advancement in decentralization of clinical trials, how we transition from telemedicine to telehealth and also expand the scope of these regulations to include clinical trial. And I'm happy to see that now DPDP would cover certain elements of this. Okay, and that's where this regulatory advancement will help all the stakeholders, sponsors, academicians, patient advocacy groups, all technology experts. Okay, and that's where I think Nandini ma'am spoke about. We need AI experts. Just to give you an example, like when we spoke about digital innovation, it can be bucketed under three elements of the clinical research journey. First element I spoke about is the patient recruitment and retention. Okay, we need to have ethics as well as regulatory guidance on that. How social media and digital tools can be used ethically to accelerate patient recruitments. Okay, second bucket is for recording of the outcomes, where I spoke about use of wearable devices, use of remote monitoring devices, use of lot of your electronic assessment like ECOAs, EPROs, and all those. Can we have certain guidance framework around this? What are the standardization methods? Okay, and we with Make in India thing, we see a lot of Indian partners, they're coming forward and developing these tools. We have no homegrown tools available. We don't have to rely on imported tools. Now, how do you foster this environment and create the framework? We should build confidence on these homegrown tools. That's the second part. And third part is how do we use digital for analytics? And that's where I'm gonna say until artificial intelligence co-powered by human intelligence. You cannot leave everything to AI. It has to have component of human intelligence. But how AI can be increasingly used? Just to give beautiful example from Pfizer's history of COVID development, both vaccines and antiviral. I think it's available in public domain. We have seen that use of AI for different aspects such as monitoring, target identification, discovery, also predicting the efficacy. We found that the screening of our antiviral came down from 6 million targets to 600 targets. And that reduced our screening time from 4 years to 4 months. Imagine, thing which would have traditionally by high throughput screening would have taken us 4 years. With the help of AI, it took us just 4 months. And with generative AI coming in, now we also use it for various regulatory reporting. And that brought down timeline for development of these regulatory guidance, regulatory submissions by 50%. Okay, which led to faster submission of various documents and faster approval and faster access to patients during the time when we all were looking up to these new innovations. And that's the beauty of AI. But it comes with its own set of challenges: ethical AI, privacy, confidentiality, safety, generalizability, interoperability. And how do we build that confidence? And that can only happen when we have regulatory framework which will help us. See, I'm part of Indian Society for Clinical Research, and we came up with this beautiful position paper on decentralized clinical trial. I think those who in the audience who are interested can refer to the ICR position paper on DCT, published in 2022. It's available on the website of the Indian Society for Clinical Research, ICR. And that has given very beautiful narrative of what was the pretext, what decentralized components are there, what needs to be done in future. So while we wait for similar amendment in our regulatory guideline, NDCT Rule 2019 will be amended in due course. ICMR has already started the process. Okay, so we will see more coming in the near future, but we don't have to wait for it. We have some guidances from nearby countries, developed countries like FDA, AMR. We can rely on those guidances as the best practice rather than just saying we don't have guidance and generally do anything that pleases us. They do what is ethically right, what is responsible, and that will lead to better use of the uh, uh, digital technology. Now, quickly switching gear to real world data. Now we see that real world data is not only for post marketing as per our earlier assumption. Okay, now we see real world data being increasingly used right at the discovery stage, early development, phase 1, phase 2, to accelerate trial feasibility. We have single arm trial where we don't need two arms to conduct the trial. We have single investigational arm, and then we use real world arm either from the registries or from some other trial pool. So that arm works like your synthetic control arm. And these interventions have reduced both timeline as well as cost of the development. And that's the reason we have such complex designs now. Clinical trial designs are already complex. We moved from traditional RCT to adaptive designs to minimal designs to now we also have these hybrid designs where we have component of real world also incorporated. And that's the reason we need equally fast-paced amendment to our regulatory guidance, and that will build more confidence to the investigators and the sponsors. Yeah.
Great. I think what I can conclude from your expert comment that regulatory frictions should not halt innovation. It should sharpen it. And when we talk about real world data and hybrid models, those who are woven into compliance, we move from resistance to resilience. So we need digital transformation in trials. And you had beautifully explained how we should collaborate, how we should become adaptive, and then regulatory modernization is also required. So thank you, Dr. Taur, for your wonderful insights. And now we move towards the next slides. Yeah, so Tripathi sir, are you there, sir? Can you hear us, Professor Santanu Kumar Tripathi sir?
Yes, I can hear you.
Okay, sir. Sir, can you please open your video so that we can see you also, sir? So, welcome, Professor Santanu Kumar Tripathi. We all know we had already introduced Professor Santanu Tripathi, sir, who is the president of Association of Clinical Pharmacologists, and he is a very senior clinical pharmacologist in this country, academic leader. So, sir, my first question to you, as a senior clinical pharmacologist, we are talking too much about artificial intelligence, but that is the requirement of this time only. So how do you see artificial intelligence driven drug development and trial optimization aligning with the ethical imperative of ensuring patient safety in our country? Sir, can you hear us?
Hello?
Yeah.
Come on. Can you repeat, please? No, I had a problem.
No, I had asked Professor Tripathi.
Oh,
yeah. Sir, can you hear us?
Yes.
So can you hear us the first question, or shall I repeat, sir?
I could, and I also it will be screen on the screen. Okay.
Yeah.
Yeah, there has been a lot of discussion already regarding the use of AI or AI-driven drug development and trial optimization. There is definitely need for trial optimization. The use of AI in discovery and also in the different development phase has started in different parts of the world. Of course, in India, we are trying to adapt ourselves, as has been evidenced by the response given by the ICMR. ICMR has already developed research ethics guidelines where AI is being used. But so far as the regulations are concerned, truly speaking, yes, the sensitization is taking place currently, but it has not yet been incorporated in the NDCT rules. Necessary amendments are yet to be made. My previous speaker, Dr. Santanu, has also mentioned that we are waiting for that, but we are at the threshold of accepting, integrating use of AI technologies into our regulatory system, as well as implementation of the clinical trial projects with help from the AI technologies. The concern is, as of now, if the protocol usually so far as the data collection, so far as the AI can really help in different layers and different levels of clinical trial conduct, starting from development of the protocol to the different stages of conduct of clinical trial, starting from screening of patients, enrollment of patients, to the informed consent process, and the data collection, patient monitoring, off-site patient monitoring, etc., at all stages. Now, in any clinical trial project, if we think of the situation in India currently, if the sponsor very categorically mentions the indispensability of AI, use of AI technologies in the clinical trial implementation, in the protocol itself, when they are submitting with the regulator, then perhaps there might be a lot of back and forth communications should be needed. And because truly speaking, in our regulations, no clear rules have been yet framed. So there might be some kind of confusion and some kind of back and forth explanation to be sought from the sponsor. But then again, if you think of that, at the implementation stage, the sponsor decides that certain aspects of management, trial management in the whole ecosystem, clinical trial ecosystem, they will be taking help of some of the AI technologies. So for that matter, I think they have to win the confidence of the local ethics committee. And it is again depends on the specific institutional ethics committee how comfortable they are to handle it. And to a certain extent, I think the first panelist, Dr. Nandini Kumar, has also touched upon that it all requires that the necessary capacity building among all the stakeholders, and the ethics committee is also not an exception. So as of now, the ethics committee constitution in our country, whether it is the prescription from the ICMR guideline or in the NDCT rules, right now there is nobody who can be considered as an expert in this area. But then again, ethics committee has been given the flexibility that as needed, as they feel appropriate, then the ethics committee can always call for a subject expert. And for that matter, the use of AI technology and to what extent it impacts on the patient safety, impacts on the patient's, impacts on data privacy, all these issues, ethical issues. So there certainly, in order to be more informed how AI is going to impact on these issues particularly, the experts can be okay. So the experts can be concerned expert in AI technology. His knowledge can be sought for, and then of course there should be also provision made by the ethics committee, as recommended by the ethics committee, how these will be guarded so far as the patient's autonomy is concerned, patient's understanding, in the use of AI. AI can impact on all the stakeholders, whether it is the investigator, the study team members at the site, or whether it is at the level of the patient also. There might be in certain situations there you can use an AI technology or some digital solution where in order to get sufficient data inputs from the patient, you want your patient also to be supportive of it, and patient is also to a certain extent would be you can say that AI informed or AI should be comfortable to handle that technology and facilitate in data entry into the AI-based database. So for that matter, I think there is also a need for sensitizing the patients or the participants also how to use the technologies at their level. So at the present, it is a challenge definitely, but then there is a time, we are at a threshold, and it is time that we gradually try to sensitize all the stakeholders and incorporate the very idea and concept of integrating AI technologies into our clinical trial conduct. At the same time, we should also think of that there are certain challenges also, there are some risks also. There are issues like algorithm bias, there are issues like data privacy, and how are we going to guard against that? To a great extent, some of these issues has also been highlighted in the ICMR guidelines. I think the current GCP training programs, awareness and training programs, in which there is a lot of emphasis is given on ethics education by the ethics committee members. This portion is up till now not being deliberated, not being discussed. I think it now demands that we should also try to educate our ethics committee members also how they will be aware of the nitty-gritties of AI use in clinical trial conduct. Maybe slowly we are getting smarter, and I can only hope in another couple of years or maybe in a year or so, we will be in a much better position. So that at the moment that I can comment. So far as from Indian context, yes, if you intelligently use AI, yes, of course, the efficiency would be improved in all respects. So far as quality of data as well as the patient safety, the two major arms that are otherwise covered in the good clinical practice. So both will be strengthened if we can intelligently use AI. So that's what I can say. AI should rather use as an intelligent tool to be used judiciously, and we should not allow the AI to be a master on us. So it should be just a technology that should be used judiciously in order to handle the conduct the clinical trials in a much better and efficient manner. That's it. Thank you.
Thank you, sir. Sir has emphasized how we should use this intelligence, artificial intelligence, intelligently. And I think he had also emphasized on this issue that when algorithms accelerate discovery, here we are talking about drug discovery, our ethics must not, rather, we should have more progression in ethics also. So ethics also should have a proper development, because AI-led drug development, the speed of science must never overturn the safety of patients. So this is very important for all of us. And now I want to ask Professor Santanu Tripathi, sir, the next question. No, please go to the previous slide. Hello. The last question I want to ask, can you reflect on a recent Indian case where pharmacovigilance failed and how clinical trial design could have preempted that? For an example, if we talk about the pioglitazone saga, once there was some black box warning, again, it was being withdrawn with a very short notice, again, we get back pioglitazone. This type of, or very recently, we have one drug known as lorcaserin, which was there for a long time. maybe 1 year or 1.5 years. Then when some data from US we got, and US FDA banned lorcaserin, after that it was being removed from India. So what is your take on that?
Yeah, we yourself have given good two examples you have cited. Both of them are very good examples, critical examples for how the pharmacovigilance has failed. By the failure, pharmacovigilance failure, we actually mean that there are risks which could have been mitigated, risks which could have been minimized, but because of the not so robust or not so good trial design, we missed that out, and thereby patient safety has been compromised. Yes, that happens, not too often, but that happens. So, and in the question itself, the answer is there. It is the clinical trial design to a great extent which can preempt and which can actually help us in reducing the probability, minimizing the probability of that eventuality of compromised patient safety, and more participants, they suffer from injuries which otherwise could have been avoided had there been a good monitoring, scope for monitoring, had there been a good risk-based or risk minimization strategy very clearly spelled out in the protocol. And also, of course, it is not enough to have that specified in the protocol. There should be also very meticulously also comply with the protocol compliance should also take place. Another example I can mention, apart from the two examples that we have just identified, and that is about that itolizumab story. We all know about the itolizumab, which is a monoclonal antibody, which was actually introduced in India, approved in India for treatment of psoriasis, plaque psoriasis, chronic plaque psoriasis in 2013. But during the COVID time, when we were actually looking for any solution, particularly many of the COVID patients, they were dying because of the cytokine storm or cytokine release syndrome, as a result of which ARDS, and people are dying with ARDS. So we were frantically looking for a solution, and somebody came up with a solution, ready-made solution, handy solution that people are otherwise using off-label, using itolizumab for this purpose, and there were some examples, some good response, but then some sponsors, they came up and they thought of to have a quick trial, okay, whereby they tried to impress on the regulator, and then in fact itolizumab was tried for its efficacy in COVID-19 induced CRS, the cytokine release syndrome, and ARDS. And it was a phase 2 study that was planned. The sample size was only 30, and the patients were randomized 2:1. The 2 for the investigational drug, that is itolizumab, and one for the standard of care. And it was a controlled trial. The sample was only 30, and the study was conducted, and it was in July 2020. It was actually 20th July 2020 when it was approved, okay, under emergency use authorization. So, but then later on there was a lot of criticism that whether it was because there were the few areas where the criticism came up. The type of approval was, of course, emergency use authorization. We must understand that there was a huge need that time. The evidence base on which the approval was given, that was again, many people have questioned that, because it was phase 2 only, phase 3 was otherwise waived, and the sample size was quite low. And although we must remember that it was not an investigational new drug, it was a drug which was already licensed for some other indication, and it was repurposed for this purpose. And but then the time of approval was a time when there was a real unmet need, and based on that the regulator failed. Of course, there was subject expert committee who were also convinced, But then the objection was why there was no mandatory phase 4 trial, post-licensure phase 4 trial was asked. The clinical trial design was also criticized. It was not enough robust. And these are some of the reasons why. And people also said that the clinical trial design could have been a little more strengthened in terms of the so-called Simon's two-stage design. When within the same trial you ask for initial few patients, part of the sample size, in which they will do a rapid testing, okay, part. And basically the whole trial will be split into two phases, two stages. The first stage is relatively smaller number of subjects where you try to see whether the optimum or desirable outcome is achieved. And if not, then don't go further. And if you get, then you go to the second stage, something like that. So it's a kind of a cautious stage, okay? But then, although truly speaking, Simon's two-stage design was not adopted, but then the DSMB in that trial asked for, recommended that the first 5 patients will be receiving itolizumab in a non-randomized fashion. Out of 30, first 5 patients will be getting that, and then after that you can go, if you are confident, then you can go to the next phase. So these are the areas where they had gone for this, and it was approved. And later on, there were also a good number of observational studies which have also been published, and the sponsor also claimed they had good sample size observational studies, also post-marketing observational studies, although it was not marketed for with this particular indication. So these were there. So at the hindsight, we can now criticize, but the situation was such that time that almost we were compelled to try, we were drastically, frantically trying to find a solution, and we thought it is a good solution. And many people also later on published their observations. Trials have been also conducted. They have shown. So this is one case you can say that where people have said, because in the same trial there were 3 dates also took place. And later on also, it was very difficult to identify whether the dates that had happened in such COVID patients, COVID-19 patients, whether the investigational product or that treatment product, whether that was the offending drug, etc., cause of the death. But then the safety of the product was not beyond question, because the quality of evidence was not very desirable. So people had questioned. The answer to this is only that the trial design has to be robust, the sample size should be adequate, and a proper risk management strategy is to be spelled out a priori, and that should also be clearly written in the protocol, and we have to take measures that is also complied with properly. So with this, I think, yes, there is provision for this in the designing of the trial, where patient safety would be put at the foremost priority, and any compromise with patient safety is not to be accepted. Okay, is not acceptable. So the message should go that way. Yes. Thank you.
Thank you, sir. Thank you very much. I think you had emphasized a very important point for all of us. Yes, every pharmacovigilance failure is a design flaw in disguise. If trials are not built to detect the right risk early, the system ends up learning the hard way at the patient's cost. And you had also emphasized regarding the accountability issues, how we should become more proactive, and how the ethics should have an innovative approach. I think we also got certain this type of controversial issues if we talk about pharmacovigilance in India and reflections on certain COVID vaccines. For an example, Covaxin, certain public media trials that had happened. So I think that also need to be avoided. And we should have, or rather than we can say, like when public trust hinges on scientific transparency, this pharmacovigilance is not just a back-end system, it is the front line of all ethical trial design. We need to explore it more. And as we have less time today, we have to move towards our next panelist. Please change the slide.
Dr. Shambo, just a word. Is there any other occasion I need to be present, or if you please pardon me?
If possible, if you can present at the end of our panel discussion, we will be grateful,
Okay.
sir.
I'll be there. I'll be there.
Okay, sir. So
Thank
please change the slide. Yeah, so we are really delighted to have with us Dr. Priyanka Bosh Bhattacharya from Abbott Clinical Governance. So welcome, madam. I had a first question to you. As we are talking about global scrutiny on trial integrity, how do Indian arms of different multinational companies maintain the GCP compliance while working on resource-constrained trial sites?
Sure. Thank you. Thank you, Dr. Shambo, and a happy International Clinical Trials Day to the delegates as well as the esteemed members of the panel. You're absolutely right, Shambo. There is an increased scrutiny on trial integrity. I mean, whether it's the US FDA, EMA, or even our own CDSC office, there are more inspections that we are seeing. There are more, sudden ways that suddenly, be it at the site level or sponsor level, where the checks are being looked at. So I think for us as within an MNC, it's very important for us to maintain GCP compliance. We have to keep things in order in the house. And what I can say is that we are taking a three-pronged approach towards this. I mean, first of all, we have to do it at the site level, as you have very, highlighted that most of the sites that we sometimes work on, just depending on the therapy area that we are working on, that there can be resource constraint at these trial sites. So it's very important that we, work with the trial site in developing, the capabilities of it. So there are two things that we can do. First is, of course, build the capability of the site. This can be done through trainings, workshops, certifications, and it's something that should not be overlooked, because there are times when we have a principal investigator who's very well experienced and renowned, and but he or she may have a team that is not as experienced. So the importance of training initiatives is very important. You cannot do away with it. So that has to be a continuous approach that we take in terms of capability building. But on that note, I would also like to highlight, there is a evolution of our clinical trial sites in India. And this I speak from my own personal experience. I've seen that over the years, the sites are also investigating, the hospitals, the institutes are also investing a lot more in developing these clinical research departments within their institutes. And, these clinical research departments are very good to partner with. And because of that, one of the other approaches that an MNC is taking is that of a preferred partner. So what happens is we work with them, we do capability building. They are also, putting in the equal amount of effort in it. And then that becomes like a preferred partner for us to conduct our trials. Because when we are conducting global trials, the global team is one team that we have to handle, and then there is the local team that has to manage. So that is one aspect. The other thing we are doing is actually taking the risk-based monitoring approach. Because initially, I mean, many years ago when it was started, there was a bit of a resistance from the sites itself, because, the 100% source data verification was like the gold standard. So it was like, how can you not do a, monitoring visit at the site and expect the data to be of good quality? But over the years, there is not just acceptance at the site level, but also from the regulations point of view. I mean, even if you look at the recent ICH E6R3 GCP update, you will see that there is a big focus on quality, and I mean, it's at the core of it, and even risk-based monitoring is being really encouraged, and there are more guidelines and clarity on that. So that is another way we are, working with the sites, because what happens is that when you do a risk-based monitoring approach, it reduces the burden at the site, because they don't have to deal with these monitoring visits with 100% source data verification. The connectivity with the site, the centralized data collection, now that helps. in reducing the burden of the sites. Then at the sponsor level, we are looking at, we have to have a lot of self inspection in place. And again, I've seen an evolution of in that field as well. I mean, in the past, I was in the operations role, and we used to have a quality person within the operations team. And that quality person used to conduct these self-inspections. But now, more and more, I see that there is a separate entity. I mean, I'm also an example of that we have a separate team that exclusively does this. And this is just to make sure that, we are maintaining high standards of compliance and quality. And those kind of self-inspections are done in a very, in a manner not to, keep an audit kind of a mindset, but to have a quality mindset where we want to keep things in order, be audit ready at any time, and do things the right way. Because this is a highly regulated industry, we can't afford to miss. And then finally, I think this is like the best aspect I've seen in the evolution of clinical research, is that we are also looking at this, from a patient level, at a patient level. So we, at MNCs, there is I mean, when we approach clinical trials, we are also looking to partner with the patient voice here. And we want to hear the patient voice while developing our trials. So we have started partnering with NGOs. And that, I think, is very important because at a personal level, I feel that, this was the missing piece in clinical research, say 15, 20 years ago, where the patient voice was not being heard. And now that we are more and more looking into that, I think that is where we are very, it's very important that you give the right education, the consent is taken properly. And that is an aspect of compliance that is under a lot of scrutiny. But if it is done with the right intent, with the right partners, I think we are able to keep things in order very well. So that's what, I mean, in terms of, managing the kind of scrutiny that we get, we have to be, taking care of all aspects of the clinical trial that we are conducting.
Thank you, madam. I think we need to acknowledge that good clinical practice is not just a checklist, it is a culture. And India's resource constrained sites, I think multinational companies must shift from compliance enforcement to capacity building, because quality can't be outsourced. It must be owned. So I think that should be one of the very important approach for our all MNCs. Now coming to the second question, madam. How is risk-based monitoring being adapted to India's heterogeneous trial ecosystem? What is your experience?
Yeah. That is again very true. I mean, risk-based monitoring. So you know what? On one hand, we are the most populous nation. We have immense potential in terms of healthcare qualified investigators as well as patients in need. But at the same time, we are also very advanced digitally as a nation. And that is the technology which is coming to the sites as well. And this risk-based monitoring approach is gaining more and more precedence because the sites are ready and it's turning into the new norm of how to conduct monitoring. And it has its advantages. Truth be told, it is a more cost-effective way of looking at the data. And at the same time, we are doing a sort of course correction and picking up the mistakes while we are working on the trial. It is not practically or financially practical to be able to come towards the end of the trial and then realize that there were too many dropouts in your patient population, or that the data that you have picked up is not adequate to come to a proper conclusion. So with those kind of thought processes, risk-based monitoring is really gaining precedence. And also now there is always, there is an evolution also of the regulation. So there is more clarity coming from the regulators. There is more acceptability of this data from the regulators as well. So how we are approaching it is, honestly speaking, the basics of risk-based monitoring remains the same. You have critical data points that you identify, or you look at high-risk sites and you take that up. Now, as you mentioned, India has a very heterogeneous trial ecosystem. And honestly, our sites change with the therapy area we are looking at. And you see a growing trend of researchers moving from the big cities towards tier 2 cities, and also everything has to be taken into account, has to be accounted for while planning this. So the two things that MNCs are doing is that, first of all, they're selecting the right CRO or the partner to conduct it, because this piece of monitoring is usually outsourced. And every CRO, especially the MNC ones, have a lot of capability, but you have to select the right CRO for the right type of trial. Because, you know, they may have centralized monitoring, which may be perfect if it's a global trial where all the data gets pulled in, or it might be something, if it's a very unique therapy area or an indication, then you may need to have more better robust data in terms of the critical data points. And the second thing that is very important while designing a risk-based monitoring plan is that you have the right data to design your plan. And for that, companies are investing, both companies and researchers are investing a lot on collating data, like creating new dashboards, creating looking at linkages, looking at how one data point may lead to another. That kind of information is very important to look at the risk-based monitoring aspects of it. So what I would like to say is that while India has a heterogeneous ecosystem in terms of capabilities, if you choose the right CRO, then they will be able to come up with a plan that provides the right kind of data points to do that monitoring. And if you have the right kind of data set available internally, then that makes it very easy for you to identify the critical data points in terms of your endpoints or in terms of the site risk involved, and accordingly you design it and have a at the end the outcome is to have very good quality data so that we can come up with really adaptable and applicable data, insights to the drug in question or the device in question.
Thank you, madam. So what I think that risk-based monitoring, it is not about watching less, it is about watching smarter.
Correct.
And India's diverse trial setting, where we are talking about heterogeneity, it is the algorithm that needs local context, not just global calibration. So again, want to thank you, Dr. Priyanka Bhattacharya, for your wonderful insights. And now may I ask, please change the slide. Slide change, please. I think we have the next question for Dr. Anirban Rai Chaudhuri, who is the head of development excellence in Sun Pharma. So welcome, sir. My first question to you, in competitive generic and specialty space, how Indian company manage trial innovation without compromising on timelines or ethics? What is your take?
Yeah. First of all, thank you for having me here. Just confirm that I am audible because I'm speaking for the first time. So just
yes, you are audible.
Great. So yeah, firstly, I'm not talking here on behalf of Sun Pharma. I mean, I'm talking on behalf of I have worked for multiple companies. I'm kind of going to share my collective experience across last 25, 26 years that I've been doing clinical trials. And the advantage and disadvantage of being the last speaker of the group is that I think everything has been spoken. So when you talk of actually innovation, now innovation is not just about AI. I mean, AI is one part of innovation, but so I'm going to talk of TI, that is, true innovation. That is, or in other words, it's frugal innovation. So can you, when you are doing a clinical trial, can you innovate in a way so that finally what do you want? Do you want the drug to come to the market faster and you have the right data in terms of its efficacy and safety? So how we can actually kind of start from the beginning of when we are actually thinking about the trial, and that is what is all about innovation. Now technology comes in between bits and pieces, but I think what you call as true innovation comes from the human mind, and that is where I think the strategy important. And so when we start with a clinical trial, I think one of the thing is today, most of the, I think we all kind of keep talking about clinical trials are getting complex. Who is making it complex? We are making it complex. Why? If you look at, there is one primary objective, two secondary objective, and there are 8 exploratory objectives. Because one clinical trial, first of all, you try to get a lot of answers, but that actually makes the trial more complex. That actually, you know, if we can simplify that, that actually can reduce your timeline, that actually can reduce your error, that actually can make the quality better. So I think that is one important, I mean, which I personally believe that trial should be as simple as possible, because with so many exploratory objectives also, the burden on the patient or the participant also increases extremely high, right? So, you know, Santosh spoke about these e-pros and stuff. Like, you know, we feel the e-pros actually make things easy, but for a patient also, it's quite a burden. I mean, some cases we actually, it happens that patient has to go for 8 to 10 different, you know, different questionnaires they have to fill, and that's something again leads to complexity in the clinical trial. So one part is the trial design. Now another, and in this context, again, you know, the various innovative designs like an adaptive design, basket design, this work really well. And I'll tell you, like, you know, this is working extremely well with all other regulators, but unfortunately this doesn't work. If we are talking from Indian context, it doesn't work. We have tried in multiple other, you know, organization. I mean, if you present our adaptive design, it gets accepted, but ACC says that, you know, you, before you get to the next dose, you come and present. So that actually defeats the purpose of an adaptive design. So yeah, so that regulatory, you know, hurdle is there. But if I look at from a global perspective, adaptive design and other innovative designs really help in expediting the timeline for clinical trials. Then comes the regulatory pathway. Again, that is where also we can decide multiple ways, right? And if it is a 52 weeks trial, we can always negotiate with the regulatory agency that for the primary submission, we will use the 26 weeks data. I mean, this case by case basis. And then the supplemental data will be the 52 weeks data. So you actually get the trial in the market probably or possibly 6 months in advance. But then that needs to be properly defined. Your endpoint has to be properly defined and then negotiated whether 26 weeks works or not. I mean, for like in a simple spell like a diabetes, it can probably work. And in certain many other cases, actually, it works really well. Then comes, as we continue through the, I think Dr. Tripathi also mentioned, through the life cycle of the study, again, it also links back to some extent risk-based monitoring. But what we do, you know, it's about, you know, continuous data evaluation, right? You know, say whatever data we decide, like, you know, a section of the data every month, or it can be every 2 months, depending on the study design, that we continue to review the safety parameters, the critical, the critical, data points, the safety data points, the efficacy data points, and that is where technology helps. I mean, you don't need very high-end risk-based monitoring tool. Even Power BI throws out fantastic analytics based on which, you know, your clinicians, statisticians, and the data management team actually can identify, you know, trends, and based on which either, you know, that can actually lead to what we call as on-site monitoring, or actually if there are any data issues that can be actually, you know, resolved through data queries also. So various ways this can be done. Similarly, like, you know, when you go to the next stages of, I mean, for the CSR writing, again, there are a lot of tools which are coming, but most of the tools are not probably foolproof. But you can actually start writing the protocol, the CSR in advance. You don't need to wait for your top line to come. I mean, even part of the work can also be done. So if you, in summary, if you really plan well, and that is what I call innovate within your framework, and try to make sure you optimize every single step so that you can get the trial faster and more importantly get the product faster in the market without even losing any insight on the on the data quality. So I'll stop there. You're muted, I think so.
So if you just touch upon about some generic space, how you are actually approaching.
So what do you mean by generic space?
When we talk about different, that the generic drugs you are marketing in different parts of the world. So in that, how you are using this type of innovation skills?
Yes, so again, it's the same thing. See, whether you are doing a study with a generics medicine or it's drug a study with a innovative medicine, the study principles remain the same, the rigor remains the same. And a particular product, and in fact, something is going on right now in Japan, which is actually generics in the US, is actually a new drug in Japan. So, or it may be a new drug in Russia. And in fact, we are doing a study with Japan and Russia, where both the countries is considered new drug, but is actually a generics molecule. So again, we don't really differentiate between a generics and an innovator. While I personally work only on the innovator space, but as far as the standards are concerned, is the same standard and same processes are followed.
That's great. I think there are certain controversies regarding the generic manufacturing, especially there are certain group of drugs where we can waive the bioavailability, bioequivalent studies in India, but not in US. So you had clearly mentioned that you are following one rule that applicable for all the different countries.
As far as the rule, basically following the rules of the country, for the registration rules of the country, the drug goes through that kind of a clinical trial.
Right. So what we can conclude from here, that in India's high-speed generic race, because we are now wanting to become the pharmacy capital of world, true innovation is not just about beating the clock. It is about bending the curve without breaking the code of ethics. I think now we are moving towards the next and final.
And to your point, from when you talk about pharmacy of the world, it is, I think that if you look at the viewpoint of the Indian government and coming with Vikshit Bharat concept, they also Indian government is also pushing Indian companies to move into innovation while keeping focus on the generics, but also look into innovative space, and that is where I think most of the other Indian companies are also moving. And you know about this enmetazobactam, which is the first beta-lactamase inhibitors after so many years, which actually came out of the Indian market, and Sridhar Narayan was one of the co-inventors. He was from Bugworks, but I think this drug came from Orchid or one of the companies. But this is the first NCE to be registered by US FDA. So this innovation is happening at the Indian manufacturers' perspective also.
Yeah, so we are proud of our Indian innovations, and I think ICMR also has launched that nafithromycin, and very recently that was again an indigenous product. So now may I ask you, this could be very much useful for our postgraduate students and those who are aspiring to prepare trial protocols. So can you share a case where a trial protocol had to be restructured due to some participant non-compliance or dropout, and what operational changes were adopted?
Okay, so yeah, I'll give you two examples. One from kind of current and one probably from the past, and one operational and one kind of from a study design perspective. So there is one study on acne with anti-androgenic agent where this is actually for the study for Japan market, and since vitamin D has an effect on androgen level. So PMDA actually asked for a particular vitamin D level from the as an inclusion criteria. I'm not getting the details of it, but there is a specific, levels there. But when we started the study, we realized that, there is a 80% screen failure because of the vitamin D level. And when it had to actually go to PMDA, and we had to negotiate with on that criteria. So we had to renegotiate on a particular level, which is lower than what they had actually thing. So we basically kind of had to get into like what would be upper level, what is the lower. So it went to a limit rather than a specific cutoff. That actually reduced the screen failure from 80% to 20%. You still have screen failure, but 20% is still manageable as compared to 80%. So we actually had to make an amendment into the inclusion criteria to get more patients into the study. From an operational perspective, this was like, years back we were doing a prostate cancer study with leuprolide, and that is when, so as per design, the patient actually has to come every week, and that is one of the requirements from US FDA, that you have to come every week for PSA and the testosterone level, and to make, to see when exactly the patient is reaching the castration level. So for a prostate cancer patient, it was actually getting very difficult to follow up every week, and that's actually a lot of burden for the patient. Even if you look at like 3 cycles, like the 3 months, then it becomes like it becomes like a huge number of visits. So what we did not change any design as such, but we made like, for the visit 1 and visit 4, patient needs to come to the hospital, but visit 2 and visit 3, a phlebotomist with a nurse goes to the patient's house. And again, this is what I call as decentralized clinical trial. You don't need high-end tools or anything. You go to the patient's house and you collect the blood, you do the vitals, and you bring it to the site. And that is when it not only helped in actually reducing the patient dropouts, but at the same time it actually increased the area of of our thing, and especially some of the areas like in UP, even like within just of 80-90 kilometers, patients were actually started participating in that helped in recruitment as well. So this is another example I could give. You're muted, Shambo.
I think these 2 examples are really helpful for those who are listening. And I think I want to summarize here like that. When trial designs clash with patient realities,
yes,
dropouts are not data loss. They are feedback, right?
Yes.
And the smart pharma listens, adapts, and redesigns with people.
Yeah.
Not just protocols, but also in mind. So thank you again. You have your comments actually had encouraged.
I want to give one more example. Okay, so
Sure,
I'll take a couple of more minutes. So I also participated as a trial participant in the Sputnik V phase 3 study.
Okay.
So it was initially for 6 months, and then they increased the thing because they wanted to see the immunogenicity level, like up to 9 months. So there was a protocol amendment, and they were like, can you come over? I said that, that is the time of basically the second wave of COVID 2022. And he said that we're very scared. We were very scared to even go out of the house. He said that, okay, if somebody can actually come home and take the blood sample, then I am more comfortable and I can sign the informed consent form. So that when they decided, they said that yes, somebody can actually come home and take your blood sample. It's about just taking a blood sample. And that actually, because of that, actually, I signed the informed consent for the extension of the study. And this is my personal experience as a participant of a phase 3 clinical trial.
Great. I think these examples are really mind-blowing. I think it will be very much helpful for our research parties, the those who are participants today. Please move forward to the next slide. Next slide, please. Can you hear us, Team Planet? Can you move towards the next slide? I think we, due to there is some issue with Professor Santanu Kumar Tripathi, sir, and I will not get him as this for this particular question. Again, I am requesting Team Planet, please do the slide changing. As previously we had a discussion with Dr. Somnath Bosu, who is a regulatory person. Unfortunately, due to some issues, he had not joined today. And he had mentioned one thing. Yeah, please hold on. Please hold on. He had sent me one message so that I can read out for all of you. Basically, what I felt during my stay in CDSCO headquarters, the hurdles and hesitation with fear of problems in the mind of investigating doctors or researchers need to be eradicated. There are a lot of prospective advancement in doing clinical trial, especially those which are sponsored by the big companies and being conducted as global arm in India. This he had sent me as message. Yes, we need to celebrate this day of International Clinical Trial Day for that historical scurvy trial by Dr. James Surgeon, the Scottish naval surgeon James Lynn. As we had mentioned, one important issue he had mentioned, that documentation is the key element, and I think we all should be agreed upon that. Documentation is one of the very key elements for all of us who are doing clinical trial. Next slide, please. So as some issues happened with Professor Santanu Kumar Tripathi, we are not getting him. So we are also moving towards the end of the panel discussion. Can you please change the slide, please? Team Clarinet, can you please change the slide? Hello, yeah. So what are the key takeaways from this panel discussion? Number one, we had discussed about innovation. So artificial intelligence driven trial optimization and decentralized trial models, they are actually reshaping clinical trial operations. And we also learned how the real world data is bridging the regulatory gaps and enabling rapid evidence generation. Next, please. So we also learn about inclusivity, focus on underrepresented groups. We had discussed about elderly population, tribal population, rural population, and these are crucial for ethical generalizability. We talk about justice, and when we talk about inclusion criteria, they need to evolve based on population health realities. Next, please. When we talk about ethics and regulation, important concepts are coming up: proxy consent, digital risk assessment, and trial explainability. These are the emerging ethical frontiers. And when we talk about CRO registration reforms, we don't have much discussion on that perspective. We will have a future discussion on that. But we had a good conversation with Dr. Priyanka Bhattacharya regarding this risk-based monitoring demand and which agile compliance frameworks. Next slide, please. Can you please change the slide? We also try to develop certain strategic recommendation. Number one, how to build trial-ready ecosystems. regional research hubs, ethics committee training, data infrastructure, and patient engagement models. Then how to harmonize different digital innovation and regulation, how we should have a collaborative framework between CDSCO, ICMR, and industry to guide artificial intelligence or real world studies which can be used in trials. So registries. Next slide, please. So next, we also try to understand. Next slide, please. The importance of ethics committee empowerment. How we can introduce different evaluations tools which should be standardized one for digital, decentralized, and community-based research. And finally, how to ensure the inclusive protocol design. This should be mandatory. We need to think about that. Mandatory representation benchmarks in publicly funded trials. For an example, it should cover age, gender, location, etc. Next. So these are few important focus area. Future, we need to focus on capacity building, upskilling the clinical and regulatory workforce in digital trials, artificial intelligence, and data ethics. Then how to ensure transparency and trust, how to promote open access trial registries, participant feedback loops, and post-trial disclosure. Then number 3, we need to promote the public health alignment, how we can align mega trial priorities with India's non-communicable disease and also infectious disease burden. And finally, the global collaboration. So position India as a hub for ethically robust, globally impactful clinical research. So these are few important road ahead for us. Next slide, please. So call to action, we must do research with the pupil, not on the pupil. Let's commit to future where clinical trials are number 1, innovative, but never at the cost of safety, inclusive, yet scientifically rigorous, and ethical in spirit and not just in paperwork. Next. Next slide. There are certain technical glitch as we were talking much on ethics and AI. I think both of these had some problem with our. Can you please move towards the next slide, please? Yeah, thank you. So what should be our role as clinicians, regulators, researchers, and sponsors? Drive a cultural shift in how we design, conduct, and communicate clinical research. Next. So this is a very important message for all of us. This had been from taken from Mahabharata, when Yudhishthir asked Vidura, how I can be a moral king. So our research should be moral one, and how we should ensure our research moral. I think this message from Vidura to Yudhishthir is very true for all of us. That is, Atmana Pratikulani Pareshuna Samachare. Do unto others what you would like others to do unto you. So as a researcher, we need to think when we are taking our patient into our clinical trial ecosystem, we need to sit in the patient's or participant's chair first, and then we need to decide whether we should go or no go with that participants. Next. Next slide, please. I think that was the last slide. And when we talk about Bharat here, I just taken this abbreviation, bridging human, animal, and environmental health through ethical research innovation. India is today telling about one health and one world. So it could be our pillars, that Bharat framework. Number one, the One Health surveillance. We had talked about zoonotic disease, antimicrobial resistance, pandemics. Then how our ecosystem will become ethical, how we should maintain the research transparency and community trust. Then global equity, the access to innovation for all, that is also important. And digital health and artificial intelligence we were talking about. So one world, one health, and one future, that is our goal. We need to empower our country, our Bharat, to lead the way in responsible health innovation, not just for our country only, but for the entire planet. So I think we had a very good discussion today in this panel discussion. I am really thankful to all the panelists for giving their valuable time and inputs. Thank you all. If someone has, our panelists have any final comment, may I request to unmute themselves and please tell us. Please stop the screen sharing. Screen sharing, please stop here. Any comments from your side? So, madam.
I just wanted to actually emphasize on the fact that ethics committee training would be required to a great extent if it has to face this AI era. That's at most, because one of the pillars for protection of the participants is ethics committee review process. The other one is informed consent, which has been addressed in many ways in all these guidelines and policy matters that have come out of India. Abroad also it is there, but we are talking of India right now. So from that point of view, informed consent, ethics committee training are most important. And the others actually will fall in place, from the scientific point of view, algorithms, on which basis it should be, what population it should be, how many diverse population it should consider, gender, ethnicity, religion, everything. That is all on the scientific side. But from the protection of participants side, I would emphasize on these two aspects. And also get the informed consent of the patient and or the participant. In everything, this has been actually emphasized, even when sharing the data. All these instances, you require the patient to give the informed consent.
Thank you, madam. Any final comments from Dr. Tau, Dr. Bhattacharya, Dr. Anirban Rai Chaudhary?
So I think, thanks, Shambo. Very insightful discussion. What I would say is that we have to emphasize more on education. And it's not only patients. I think we as clinical research professionals, each one of us, every stakeholder who is involved in this journey needs to be trained, retrained till we achieve the success of all the clinical principles. I think that's gonna be the success mantra for all this, because it is an ever evolving landscape. We cannot just rest with our knowledge, which is so dynamic. Thank you.
Thank you, Dr. Tau. So Any comment from Dr. Bhattacharya and Dr. Aishodri? Okay. So we are ending to today's program. We had celebrated the International Clinical Trials Day. We are really thankful to all the faculties who were there today since afternoon, and on behalf of Physician Research Foundation West Bengal branch, under the aegis of Association of Physicians of India, West Bengal, and our co-organizers, Indian Pharmacological Society, Forum of Ethics Review Committee of India, FERCI, and Association of Clinical Pharmacologists. I want to thank all the delegates who were there since 2 p.m. and it is almost, we are approaching towards 8 p.m. now. So really thankful to all of them. Now almost we can see more than 300 participants are there. So we are really overwhelmed with your responses and hope we will organize more scientific sessions like this. And we are really thankful to all the faculties who were there today. Thank you all. Thank you very much.
Thank you. Bye.
Bye, 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 bye.
So thank you so much everyone for giving us the opportunity to host this session. Good night to all of you.